Vidya, Vidya Magar, can you start your presentation, please? Vidya, can you hear me?
Recording in progress.
Yes, I'm. Yeah, I have already joined. Slide already have shared.
Is Dr. Pradeep Paik. So Dr. Pradeep Paik is Associate Professor from School of Biomedical Engineering, Indian Institute of Technology, BHU, Varanasi. So his research interest is materials of healthcare and nanotherapeutics. He's Associate Professor and he has completed his postdoctoral research from Israel and Singapore. So he is currently working on translational nanomedicine, therapeutic technology for drug and gene delivery, cancer therapy, nano vaccination, immune therapy, etc. He has led more than 23 patents, 100 research articles, and couple of book chapters. He is recipient of many awards such as Outstanding Scientist Award, ISCA, Young Scientist Award, Swarna Jayanti Endowment Fellowship, Associate Fellow, member of National Academy of India, NASI, visiting scientist faculty at Israel, Rice University, USA, awarded a number of research paper, cash awards, best thesis award, and number of best oral poster presentation awards. Presently, 10 PhD students and 2 postdoctoral scholars with 17 group members are working with him in the Indian Institute of Technology, that is Banaras University, Varanasi. He is also a member of various scientific communities and serving as editors and associate editors of various scientific communities. So I am privileged and honored to invite Professor Pradeep Paik to deliver his talk. Dr. Paik sir, could you please start your presentation? You're muted, I think.
So good afternoon. Whether my voice is coming there?
Yes, sir. Yes, sir.
Yes. Good.
Thank you, sir.
Good afternoon, all. Good afternoon, all. Thank you, Professor Jore, for the nice introduction about me. I have seen that it is already delayed, so I'll be taking 35 minutes, 40 minutes. Is it okay? What about the time allotted to me for this talk? It's so? Okay. Thank you very much for the invitations and giving such a nice introduction about me. Good afternoon, all these participants here in this conference. Okay, now see whether my slides are visible there. Can anyone respond to me?
Yes, sir, visible.
Okay, whatever I have decided today is it's quite unprepared actually. It's so busy life. I could not come. Professor Jore and their Vice Chancellor invited me, but due to some other academic activities and administration work, I could not spend 3 to 4 days in this time. Anyway, so let's see what I have planned today to give some flavor about our research work, whatever we are doing for the materials used for the therapeutic applications. Okay. So title of this talk is Polymeric Nanomedicine for Sustained Drug Delivery and Therapeutic Applications. It's complete, clearly visible. You see this, somebody had some confusion is that what is the BHU and IIT BHU? So BHU and IIT BHU are, this is the in front of campus, some part of the campus, which is only shown the IIT BHU part, okay? BHU and IIT BHU is completely separate. So we are engineering, now we are in the IIT. Okay. So my lab name is Materials for Healthcare, Nanomedicine and Therapeutic Applications. It's not coming. Okay, next research level. Materials for Healthcare and Therapeutic Applications. Okay, what we are doing? We are doing polymer-based nanotechnology and biomaterials, delivery systems, modeling, designing and development of effective long-term delivery systems, insulin, anti-cancer drug delivery, different type of gas delivery, okay, insulin, growth factor, gene therapy, agent, development of vaccine, and let's say MTV-related treatment. Okay, we synthesize the biodegradable polymeric delivery system. Ultimately, we should absorb by the body. We create new biomaterials. They can behave like a memory. They can behave like a surface switching materials. It means that they have some different surface switching property. Okay. We also use carbon-based materials. We synthesize them. So believe us that we never purchase the materials, whatever actually we use other than the precursor materials. Our materials, we qualified them for the biomedical applications. Okay, till today, whatever we doing. So our group used polymers, ceramics, metal composites, so all domains of the materials. We functionalize them in terms of the pores, in terms of the hollow mesopore structures, okay? And we conjugate them. Now we are doing animal trial and waiting for a few materials, whether we can do some clinical trials. Okay, who joined with us? So chemical engineer, life science people, chemical, biochemical engineer, physical sciences people. So all communities of the students, collaborators work with us to achieve the goals of the products, to use them for the healthcare application. Now start with the story, with the lab story. Okay. So whenever we are talking about the delivery of these materials, whether it will be targeted. Okay. Let's start with the ancient story, lab story. Okay. So lab story started with heart to heart. Let's say someone already fall in love with someone. Okay. How to send a letter. How to send a letter. And ancient days, what happened? The letter put inside this envelope. They send to the pigeon, give the pigeon. Okay, pigeon carry a particular body. Let's say pigeon go and sit on the, let's say, Okay. And found that there are a lot of girls, beautiful girls
Could
are
you-
there. And among them, one is princess. So prince sends this letter, but if the pigeon is not properly trained, and if the address properly written on the letter, then what will happen? Pigeon will not be able to detect who is the queen, okay, and who is the princess.
Oh,
Yes, my boss.
yeah. Could you please put your screen on full screen mode?
It's already full screen.
It is full.
It is full screen only. It is full screen.
Okay. All right, sir.
It's
Okay.
to me here.
Okay,
I
fine.
am using full screen. Okay. So that's confirm me. My voice is coming clearly there. Okay. So what happened if you have not designed, you have not address written properly, you have not trained the pigeon properly, then later will be reached to the wrong person. So that actually is this the method to assign, direct, functionalize the letter envelope with some address so that it will be reached to particular princess, okay, not the other girls, okay, or who associate person. So you have to design in proper way. Okay. Now let's say what other example of targeted delivery. Okay, let's say from Bangalore. From Bangalore, you wanted to send some city to some letter to the Delhi. What we should put? We should write the address properly. We should write the PIN number, and we should draw off in the earlier mode the letter in the letter box. Okay, let's say with the PIN number of Delhi, it will be reaching there, and by seeing this PIN number and name of the address, okay, the postmen will sorting out them and will carry this letter to the targeted person. Okay, this is the example of the target. But what actually default occur? Let's say my email ID has been given here, type.bme@iitbhu.ac.in. Whenever you will be typing, if you missed or if you miss to type a letter, or if you do something mistyping, then this email will not reach to me. Then it will miss the target, and instead of reaching to me, it will be reaching to others. So if it is a secret message, then what will happen? It will reach to the wrong person. Okay, so this will miss the target. Second way, let's say phone number has been given. 8500109932. Whatever the number is given, you type wrongly any number, then what will happen? Cross connection. You will be connecting wrong person. Okay. Then let's say you have spelled something who is supposed to other person not to know. Whoever is the owner of this email ID, it should be reaching to them. The call should be reaching to them. So these are the actually mis-targeting, the failure of targeting, failure of the message to reach to the targeted person. So these things are the example how when the we whatever we send to someone, or similar way, if you send the medicine, if you do not design them properly, then they will mix the target. So these all are the example when we can reach to the target and when we mix the target. So whatever medicine used, okay, free medicine, then what happened? Other than the targeted position, they react with the other cells. Okay, they kill the other cell. You see how? Let's say drug delivery system. It's a drug delivery system, customized delivery system. Okay, you encapsulated drug inside this. Okay, let's say target is the cancer cell or something else, or some immuno cells. Okay, some cells which will increase the immunity of the body. Okay, if you do not customize the cells or the capsules properly, then it will not reach. And another cases, let's say chemotherapy drugs kill cancers, okay, and it simultaneously also kill the healthy cells. Okay, so this is a capsules, it's mistly designed. Then what will happen? Let's say one is cancer cells, another is healthy cells. So if your capsules has been designed wrongly, then what will happen? It may happen that it will be targeted, target the cancer cell, but simultaneously there will be a huge chance. There is huge chance to hit the healthy cell also. So instead of getting the positive response, you can get simultaneously the side effect of these design of the treatment. Okay, so you have to design this properly. Now, what is that? Disadvantage of the delivering of the free drugs. What happened actually? Most of the drugs, okay, whenever they are free, not only they react with the infected cells, they react with endothelial other cells, okay, and create the side effects. Other cases, what happened? Many drugs molecules are light sensitive. Okay, let's say you have given the injection of the drugs, the patient is now out to the sunlight. Then what will happen? Due to the sunlight, it will be creating, this drug will be creating, let's say, some radicals or other reactive components, which will be reacting with the other parts of the body. Okay. And what other things can be happen that free drugs, when circulate throughout the body, there is a high chance of excreting through the urinary tracts. Okay. So you have to always use the maximum doses or more doses, whatever they require for the treatment purposes. Okay. So they are not safe. They are not safe for the healthy body. So along with this, the kill or sensitizing the infected cells, they will be infect to the healthy cells. So you have to design the drug delivery vehicle, or we have to design them properly so that we will reach to the target. Okay, since I have mentioned that nanoparticle-based therapy, then let's say what is the translocation of the nanoparticles? And let's say you have injected the nanoparticles, they will be reaching heart, brain, kidney, lymph, fetus, liver, or urine. But whether all the nanoparticles will be reaching to this particular position wherever the target, it may not be. You see how it's happened. So whenever, let's say there is some tumor, okay, of size. Okay, let's say in tumor is there somewhere in the heart, okay, or somewhere else. Enhance uptake for particular with size between 30 to 20 nanometers will be there. Let's say if it is reached to need to reach to the some lungs. Okay, let's say in blood, clearance of small particles is much lower than that of the larger particles. Let's say if it has to reach to the liver, then rapid clearance by the phagocytosis uptake and hepatic filtration for particles of size 10 to 20 meters is suitable. Okay, so significant sizes of the particles is always required. So some sizes of particle, if it is required to reach to the spleen, so there should be around 200 nanometers. And for the kidney, so kidney, whatever the filtration system is there, if the particle size are smaller than 8 nanometers, then what will happen? That will be going out to the kidney. So if you load some drug molecule medicine in the nanoparticles is smaller than the 8 nanometers, they will be going out easily. So they will not repeatedly circulate inside this body. So the treatment efficiency will be decreased. Now see, why nano is important for the biomedical application. Let's say one example is given. So different sizes of nanoparticles are there. You inject it to the blood vessel. They're going through or they're passing through the blood vessel. So there will be endothelial cells. If cancer generate, tumor generate, then what will happen? They create some pores in between the endothelial cells, and that pore size is almost 100 to 800 nanometers. Okay, so if you use the bigger sizes of nanoparticles, then what will happen? They will not cross the endothelial barrier, okay, cell barrier, and they will not able to reach to the cancer targeted site. Okay, so then what happened? You need to take the smaller sizes of particles so that they can cross this barrier and can reach to the cancer cell sites or the tumor site. So these things have to keep in mind. Now how you will be designing the nanoparticles? This is mostly general initial part, then I will be going directly. How nanomaterials reach to the target? I hope many of you know that the scavenger receptor mediated SR mediated phagocytosis can be there, pinocytosis can be there, caveolin mediated endocytosis are there. So how and what type of this endocytosis or the passing through the cell membrane into the cytoplasm and then reach to the nucleus, that's depend on the charges of the nanoparticles. So you have to keep a total idea there where the target and what is these possible charges at that position. So overall, you have to keep in mind. Now, another barrier is that whenever you will be delivering the nanoparticle-based medicine, then what will happen there? There will be lot of organic components, not cellular components will be there. Their functional properties also will be there. So whenever, let's say from the cell membrane to the nucleus, they will cross, then there will be so many, the Golgi body, mitochondria, and other things, cytosol. So if you check all them, each and every point, they are different in pH range. So pH range will be another barrier to destroy the activity of your nanoparticles or nanomedicine to reach to the target. So that thing also we have to keep in mind. Now, surface, another one, and the first layer will be coming surface charge of the cell membrane. Cell membranes will be negatively charged. So you have to design it properly in such a way that it will reach to the target site and properly. Now, so we can proceed. Okay. So we can proceed. Nanocapsules, mesoporas, polymers capsules. I will be talking polymers and few polymeric. Okay. You can make hollow mesoporas. Okay. Why we can make the porous structure of the polymer capsules? That also I will be telling very quickly. Functional properties of nanocapsule. What is will be the organic moiety, organic functional group, whether they will lethargic in transfer of the mass of the drugs or not. That also you have to. Whether your particles will be chiral or not, because chiral, our body is symmetric in nature, because most of the what is the organic component used for the construction of our bodies, most of them are chiral. So chirality make the symmetry, and chirality of the drugs also create the targeted treatment. Okay. Now, drugs, memory, sensing, and loading. So whether your capsule can load the drugs, whether they can memorize the drugs, whether they can sense the drug, and what is the loading property also will be there. Also have to keep in mind. Need to functionalize of the nanoparticles for drugs or gene delivery. Otherwise, the gene or genetic components or similar type of components cannot be absorbed inside this. Okay. Problem associated with the functionalization. Variation in the loading efficiency, problem of uptake of drugs or genes, lethargic mass transfer to the targeted site. So non-uniform size of the capsules will always disturb to calculate or to decide the doses. Okay, and selectivity problem will be there. So there will be various method will be there. I will not be going. That will be the textbook things. So say mostly useful procedure are emulsion method. They are mini emulsion or micro emulsion, which can give you the smaller size of polymer particles. So let's see how we can design. General structure is that if you can make core cell type, you can make solid core or the cell will be there. If you take the hollow core, then what will happen? Inside the hollow core, you can load the huge amount of drugs, medicine molecule, let's say, and the cell will be protecting them. So whatever I have told that whenever they will cross the endothelial barriers, okay, the drug molecules will not come easily, or it will not coming out easily, and they will not react. So you can prevent the side effect. Okay. So there you can design various type of core, single core, single cell, okay, double core, various layer of the cells you can make based on what type of drugs molecules are there and what will be the target. Okay. So I'm not going all these things. For the polymer particles, you have to take, let's say one core will be there. You have to have a monomer. You have to make a layer. Okay, monomer reactive surface. Another layer you can make monomer 1, 2, monomer 1 will be there, and another 2 also will be there. So they will be protecting. How will be protecting? By making the layer, you can actually monitor the sustained delivery, or your delivery will be sustained, or it will be burst release, or it will be fast release. That you can. Okay. So another cases, you can make you can use the cross-linking agent on the surface. Cross-linking agent actually anchor these drug molecules inside this capsule. So various things will be there. One example is giving, there is a silica. You can functionalize its amine group also. Okay. So this amine group actually bind the drug molecules. Okay. These are the general one. Now see, this is from our research. Okay. We have found that there's a surface roughness create a big role. Okay. If the particles are very rough in nature, then inside the rough, or whatever the pores will be there the drug molecules can be entrapped. Okay, so these nanoparticles or assembly of nanoparticles are formed by self-assembly of smaller sizes of particles. Let's see the second step. In third cases, what happens? Allowed the further polymerization. When drug molecules are there, anchored or loaded inside inside these pores, then if you allow the further polymerization, then what will happen? They will create the layer on the drug molecules. And whenever you inject, this first layer will be great. So let's say there will be a time after the injection to reach to the target. Then if it will take a time. So that time helps the first layer. Okay, or in between that time, the drug molecules will not coming out. Whenever it will be attaching to the infected cells, then the first layer will be degraded, and then whenever it will reach to the target, then it will start to the release of the drug molecule. See, loading of the drugs molecules. Okay, there is a solid particle. So let's say there is a solid particle which have some surface properties. If the drug molecules load properly, then let's say fluorescence add, then what will happen? It will, you can see from the microscope how the fluorescence green emission is giving in similar way. So the deep green fluorescence will be there. If your drug is hydrophobic in nature and your is hollow in nature, then what will happen? The drug molecules will be there on the surface of the particle. So if the drug molecules are there on the surface of the particles, then what will happen? It will release easily. Okay, it will release easily. But if you take hollow particles, okay, if drug molecules are encapsulated in the center, then what happens? That's a polymer layer. Okay, layer by layer deposition on the core. These drug molecules are encapsulated inside. Let's say they are fluorescence tag. Then if it is there inside the cores and huge amount of drug molecules are there, then they will give this very deep fluorescence. Okay, here it's tagged in such a way that it is giving the red. Okay, now you inject this. If you have functionalized in, then what will happen? They will go to the cell. Okay, they will cross the cell membrane, then they will reach to the cytoplasm and then to the nucleus. Or if it is required, it can go to the nucleus also, inside the nucleus also. Okay, so this designing, okay, cross-linking of the polymers, okay, this case is very important for the drug delivery application. Now I see how you will be maintaining the pores. Why you will be maintaining the pores? I believe that few of the previous speaker already talked about the drug molecules, since about how they work, what is the drawback. But let's say if you create a mesoporous or porous type of structure, then what will happen? There will be D radius of the pore. Let's say you have ibuprofen and gentamicin, alendronate or amoxicillin. They will have proper size. So if they have the proper size, then that drug molecules will be entrapped inside this D. Okay, so they will be entrapped inside the D. They will, as if they will be safe inside the pores, and they will have barrier. Okay, they will not allow easily to interact with the unwantedly to the other parts of the body until unless it is reaching to the target. So there is a role of the pores, there is a role of the surface structure of the pores. Okay, so you have to do it. Now, let's say this is from the core shell polymer nanoparticles we have designed around below 200 nanometer. You see this, how the uniform sizes are there. This is made up of polycaproelectron. You see all the uniform sizes. So if you have the uniform sizes of the polymer particles, then you can load the drug molecules each and every capsule in equal amount. And if you can calculate, the diameter, and if you calculate how much extent of the drug molecules are loaded for all over the particles or for total particles, then what you will be able to do quantitatively, you can estimate how much amount of drug molecules are loaded or medicine are loaded in a single particle. So it will be very easy to decide the doses by this method. Okay, let's say one cell will be accepting 1 nanoparticles. It will be containing few hundreds or few numbers of the drug molecules. So few numbers of the drug molecules will be entering inside the cells and it will be releasing inside the cell. Okay, now see this, how we can vary, you can vary the thickness. What are the capsule it is showing? Let's say 90 nanometers. This is quite thin in size. Okay, the hollow space, whatever showing there is dark molecules are entrapped inside this, around let's say 120 nanometers of the hollow core, and some other particles are there 32 nanometers. You can vary. Again, I will be showing is that. Okay. You see that here, this is a special type of polymer nanoparticles where it is a hollow structure and porous in nature. You see that this case is almost 20 nanometer thickness. In the second case, as you see, almost 200 nanometer sizes of capsules. Okay, it's a nanomedicine. Let's say after loading is that almost 5 nanometers thickness of the wall are there, so that this wall also can tune the release of the drug molecules and decide the doses and how much doses is required to deliver at a particular disease or to the target that you can decide. This is a special kind of nanocapsules. You see that only 2-3 type of doors are there, other than there is small ports. So one door, what will happen? So through this door, the drug molecules can enter inside this cavity, okay, and other door. We minimum need 2 doors, so that from one side it will be loading, and if there is some airy things or some other liquid, they will be going out. Similar way, whenever they will release by the diffusion of something, the force will be exerted here to release them. So this is like that. Say you are entering through door or entering through the door. Let's say your window is open, then there will be thrust, something will be coming and some air will be going out from the other door. Okay, in the similar way, your drug molecules will be there and they will be releasing and properly. Now, so this is also an example where the pH, these particles are very much sensitive to the pH. Okay, sensitive to the pH. It means that we change in the pH, the release rate is different. With time, then what is happened? pH, the capsules, and with time they will be releasing as if it is pH clock. It is like a pH clock. Okay, with time and pH, they will be releasing different drug molecules. Okay, now directly go another example. There is a polymer, it's called polycaprolactam. It has huge use in the tissue engineering. Okay, it's a biocompatible. It's a FDA approved. It easily can make a fiber like, let's say, electrospinning method, and it's used. But what we have done that, we have made nanocapsules of 200 nanometers. Where you see the whites, many spots are there. White spots are there. So whenever the white spots, actually these white spots are the pores, mesopores, 2 to 4 nanometers. So this 2 to 4 nanometers actually the channel to allow the drug molecules to enter inside this cavity, hollow cavity. Okay, so this will be there. So this channel on the surface, the pores on the surface is like a channel allowing the drug molecules, and inside the hollow cavity, this is the reservoir of the drug molecule. So you can preserve a long, and you need not have to give the much more doses repeatedly. Now there is a method of preparation. So we have taken first silica, then we have taken a soft templates, and we allowed the polymerization. What happened on the surface of the silica nanoparticles? There are layer by layer deposition of these polymers, and the short template micelle anchor inside the polymer chains. You see that these micelle are there. They are anchored or entrapped inside these polymer chains. Now what we have done, second case is when the particle has been formed, okay, we allowed to wash out the soft templates, these micelles. Then they create the pores on the surface, and I see white pores, spherical pores. Second case is we have removed the silica by etching. There is a etching method will be there. Okay. So by this way, first step we have made the core, second step we have made the hollow core, whatever there. Okay. So what have done? We have used these things to load the fluorescence dye. Okay, we have find that this red, whatever is that, this nanoparticles is absorbing or loading the small biomolecules. Okay, the biomolecules. So what we have done, we have loaded, I will be showing you that we have loaded these things with siRNA. Small interference, ribonucleic acid, okay? And then we have challenged to the skin cancer cells, okay? This in the first step, whenever we have checked this, that whatever we have prepared, we have done the treatment of the nanocapsules, they are very much safe. They are almost 90% biocompatible, which is viable. Okay, they are viable in nature. Then whatever then, whenever we have loaded with the siRNA molecules, then we have loaded, we have challenged these nanocapsules, or you can say nanomedicines against the skin cancer cell, and the siRNA was tagged with the green fluorescence protein. Okay, then after 24 hours, we found that, okay, we found that the cells rupture completely, cancer cell rupture completely, which was not observed when we treat these things with the naked or empty capsules. Okay, so you see that green fluorescence intensity is decreasing with time. Actually, it will be telling how much nanoparticles concentration you need to use to kill how much extent of the cancer cells. So what happened? Validate these things or to the hypothesis whether these nanoparticles loaded with siRNA carrying these things inside the cells or not. What we have done, we have challenged the free siRNA with the GF, okay, green fluorescence protein. We found that the most of the green fluorescence protein at siRNA are covered on the surface because it is giving on the ring pattern. It is not inside the cells. Okay, so these act siRNA molecules are attached on the surface. They are not going inside the cells. Okay, now whenever we have loaded them inside these nanoparticles and we have challenged against the cell, we found that this green fluorescence is coming throughout the cells. This is the light chain imaging, okay, of the cells. Okay, skin cancer cells. You see that green fluorescence are coming from throughout the cells, from cytoplasm, from nucleus. Okay, so these are the treatment efficiency. Whenever you have used almost microgram, how much microgram? 500, 600 microgram, and a huge loss of the intensity of the green fluorescence. It means that it's killing the cancer cell. Okay, so I'll be telling quantitative value. Oh, I have forgot the quantitative value. What happened in that? The siRNA gene silencing. Okay, what happened? siRNA entered the cell via the lipid-based transcription. Okay, then it's produced RNA-induced silencing complex. Okay, RIS. Then these siRNAs go near to the mRNA. Okay, messenger RNA, and it's hit the messenger RNA and destroy the messenger RNA. Okay, target to the mRNA and cleave them. So mRNA are responsible, let's say, for the cell division, cancer cell, creating the cancer. Okay, so if you can destroy these things, then you can stop the division of the cancer cell. Okay, this is the procedure. So if you take these things with the metallic nanoparticles, okay, and allow them against the cells, you will find that it is going inside the cell. Okay, these black spots are not coming from the polymers. Okay, it's polymers carrying the metallic particles inside the cell, and this TM image completely showing that they are going inside the cells, not only cytoplasm, they can go inside the nucleus also. These are the validity, okay, validation that your nanocapsules are going inside the cells or nucleus or not. Okay. Another thing I will be telling is that how we can make the chirally active nanocapsules by some method. Okay, let's say mesoporous polymer nanocapsules, chiral drug sensing and application. What is the importance of this? So there is a drug, is the thalidomide. It is used for the morning sickness. Okay, for pregnant ladies. Whenever it was used, let's say another 15 years before, the 10,000 patient has been treated initially, and it has found that 10,000 mother giving birth of this type of lymph baby. Okay, they are somebody has head is not there, the leg is not there, somebody has 6 fingers, so many fingers, hands are not there. So this type of defect baby are giving birth. What happening actually? This thalidomide, it have the optically active. Okay, optically active. It can be chiral. Okay, it can be RS and S type of structure can be there. R and S type of R structure is sedative and anti-anesthesia agent, and the S type thalidomide teratogenic effect give. So what happened that they directly go to the womb and to they interact with the DNA, and they disbalance the DNA and create the this type of defective baby. So genetically modified, okay, these zygotes or whatever, these small assemble of the cells whenever just it has been growing as a baby. Okay. So this has been approved by this lady in Germany, and she has been allowed to do the job till her age of 90. Okay, and this news was there in India also. The thalidomide cannot be used for the anti-NSA purpose or for the morning sickness. Then what actually we have done is that what we have done. So we have synthesized a block of polymers, chiral block of polymers, which is made up of ethylene glycol and let's say glutamic acid, aspartic acid, or phenylalanine. So they will have L and D. So ultimately, we'll be getting polymers. Let's say I am not going details what is the synthesis procedure. Their property exactly similar to the protein chain or lipid chain. Okay, they change their structure. They give the secondary structure. Okay, so they can give the secondary structure, and whenever the temperature and pH will be changed, their chirality exposed. Okay, then using this thing, okay, how much chirality, how much structure are there, we can calculate that by CD method. Okay, so that's a CD spectrum. By calculating how intensity, okay, we can calculate how much alpha, beta, gamma structure is there, and whenever they will be the coiling in nature, okay, they will be folding in nature, then how the chiral space will be exposed. So what will happen with that? So what happened? Let's go there. So we allowed the synthesis of nanoparticles, then we made the templating with this chiral block of polymers. Okay, there will be micelles which will be attached. This polymer micelles will be attached on the surface of the nanoparticles. Then further we have allowed the growth of the nanoparticles. So you see that this stage, so nucleates are there, the templates micelles are there, which are chiral block of polymers. Then allow the synthesis, allow the deposition of this nucleus, smaller nucleus. So deposition of very small particles. Then what happened? We have taken out these templates. Okay, these templates. So ultimately, when the templates went out, then you will be getting, okay, this type of mesoporous structure on the surface. Here I have mentioned the chiral, okay, mesoporous. Chirality is that in terms of the void space. Those who are chemists or biochemists or life science people, we will know that chirality can be generated mostly in organic molecules, and they are optically active. When the plane polarized light fall on the chiral molecules, they rotate according to their orientation in three-dimensional space. So they are stereocally active, stereochemistry matter is there. So they are stereoactive. Okay. So these chiral mesoporous polymer capsules we have generated. What is there? There will be polymer particles, and the pore are generated which are impregnated with the chirality. So imprinting of chirality is there. So chirality is imprinted in terms of the void space, not in terms of the organic moieties. Okay. Now question is that whether pores are chiral, because we everybody know, everyone, those who know little organic chemistry, asymmetric synthesis, okay, we know that the chirality of the molecules generate, okay, due to the chiral structure, organic moiety, but this hollow structure can be chiral or not. What we have done that, okay, let's say, what is the mechanism, then I am going. So what happened? So the templates will be there. Then we allow the deposition of the polymer chains on this. Okay, on this, you see the structure. So there will be micelles. On the micelles, there layer by layer deposition of polymers will be there, and ultimately we have removed this. Okay, so this by this way the mesoporous structure has formed. Now see how we will prove whether the pores are chiral or not. Then what we have done, we have extracted these organic templates, chiral templates. Then we have done a lot of experiment of NMR, FTIR, okay, other chirality method, okay, whether everything has removed or not. We found that there are no block of polymers are there in the network structure. Now we have to prove the chirality of the pore. So what we have taken, we have imprinted these capsules with the L chiral block of polymers, and another set we have imprinted with the D or SRNR. Then what we have taken, we have L-valine and D-valine and mixture of racemic mixture where D and L are there. Then whatever the capsules has been imprinted with the L, then we have put the solution of L-valine. We found that After certain time, the L-valine concentration decreased in the solution and they deposit on the pores, okay, on the surface. Then what is the significance is there? There will be a chiral interactions between the L-valine molecules and the L imprinted this in the network structure. So similar way, whatever we have imprinted with the D, okay, then we have put them with D-valine, okay, organic chiral molecules. It has been found that D molecules are absorbing on the D chiral. Now, keeping in the mind of the in this concept, if you have a drug molecules which you need to deliver, okay, which have the D chirality. So D chirality molecules you can load easily inside the pores. You need not have to give other pores other than the chirality. Okay. Our experiment we have done, we have taken a mixture of D and L, okay, racemic mixture. Then we put the L. And that cases also we found that only L molecules are absorbed, D molecule are excreted. They remain in the solution. So what is coming that in mind? Okay, what conclusion can be that if you have a drug molecules of racemic mixture like thalidomide S and R, they are mixture because they synthesize in the asymmetric synthesis method. Asymmetric synthesis method always give the racemic mixture, but you need to deliver R. Then you imprint with R, the R drug molecules will be loaded inside these capsules. So your capsule is now formulated with the R. So it's like a printing. It's like a printing of chirality of the network structure, okay, a chiral structure in terms of the void space. So simultaneously, what is happening? It's like a chiral memory. It can memorize the chirality of drug molecules. It can recognize, and you can load these chiral molecules inside these capsules to form a nanomedicine. Okay. So by this way, the chiral molecules can be separated from the racemic mixture. You can get a proper kinetics, how it will be there, with time, how much you can load it. So this is a formulation. So when this formulation is there, now you deliver this, so you can avoid. So we have hypothesized and proposed, some work is going on, all these cases. So this is actually the concept. Now you see the Sejoplex. Sejoplex is a concept of making, there is a term is called polyplex, polymer and genetic component. If you make the complex, then the term is called the polyplex. Okay, now here we have given a new name is Sejoplex. What is Sejoplex? SiO, which is loaded, SiO loaded with the zinc. Zinc is a photonic crystal. You see that this is very coagulated structure, but whenever we have taken the TM, high resolution TM, you see that a lot of pores are there. Okay. Whenever we have enlarged these images, we found that inside these pores, there are a lot of photonic crystals like zinc oxide are there. Okay. So whenever you have impregnated with zinc oxide, what is happening? What is happening in the system? Now see that. What happened? We have taken the DNA molecules tagged with ETRB. Whenever we have given the solution of the plasmid DNA tagged with the ETRB, we found that on the pores only, on the pores DNA molecules are attaching. DNA are negatively charged molecules. Okay. On the pores, the DNA molecules are attaching. How it is attaching? You see very surprisingly. When the pores are generated only in the silica matrix, DNA are negatively charged, silica is negatively charged. So negative and negatively charged, there will not be any binding. And there is the pores, surrounding of the pores, there are photonic zinc oxide. Zinc is positively charged. How zinc is positively charged? Zinc release the oxygen. Zinc diffuse the oxygen. There is a lot of oxygen defects will be there. So this is the defect, concept of defect will be there. So atomic defects will be there, or point defects will be there. So due to the defect due to the diffusion of the oxygen, there will be creating a slight positively charged, D plus, okay, and DNA will be negatively charged. So due to this, okay, they can load these things, okay. And the other way also, whenever the zinc oxide are there in the center of these capsules, okay, huge of the DNA molecules can be loaded. So by this way, your formulation is ready. And other things are there. Treatment procedure is similar. Now, so what is that similar way? We have made lot of nanocapsules. We have lot of uses will be there based on that. This is a nanocapsule. Let's say I have, let's say 7 minutes in my hand. Okay. This is a self-assembly capsules. Okay, it's looking like a castor apple. Okay, so this has been made. Then what will happen that these castor apple type of self-assembly nanocapsule are loaded with DNA or anti-cancer. So why I am tracing the loading of the drugs molecules? Because if you can load successfully, then your treatment will be successful. Okay, if your capsules is targeted properly. These cases, anti-cancer drugs, chemotherapy drugs, and DNA molecules are loaded. Okay, so lot of mechanisms will be there. Why it will be loading? If you know, it may be happen that somebody telling that we cannot load the drug molecules inside our capsule. No, there is a chemical structure, surface property, create a huge roles, okay, for loading these things. Here, doxorubicin are loading inside these nanocapsules. Okay. Then another set of polymer capsule, you see how it's looking like, okay, it is made up with the polymers. Its nanocapsules formulation, you see the ports, actually it can challenge in the various type of cancer and normal cell. It's found that it can target only cancer cell and it has very less effect. 10%, 5 to 10% effect will be there on the normal cell, but other 90% effect will be there on the cancer cell. So it's a cancer targeting cell. Okay. So we have done a lot of imaging and we have studied facts, other things, how we can monitor the cell division rate, okay, and how control the cancer growth. That we have also done. We have done and we have seen a lot. Last concept, I will be showing that we use the cowpea mosaic virus to design the nanocapsules. Cowpea mosaic virus will have a diameter of 36 nanometer and 64 active sites will be there on this shape. Okay. So using this, that we can template the cowpea mosaic virus property inside this nanocapsule and it will create the cavity. You see that how the nanoparticles will be there. So nanoparticles of almost 50 nanometers, thickness will be almost 10 to 14 nanometer thickness and hollowness will be around 35 to 36 okay, nanometer, exactly with the diameters of the cowpea mosaic virus. Okay, that also has been formulated with various drugs and been challenged with various cancer cells and found that they are very much effective for the treatment. Okay, so we have done a lot of things. Okay. Another one and concept has been given that whenever we have made the nanoparticles loaded with the, let's say, immunomodulatory drugs, okay, there is a response to the T cell and B cells will be there and very successful. Okay, so this response, we have loaded these nanocapsules with the OVA protein and we have found that, okay, we have found that they can be used to develop the medicine, okay, nano vaccine, okay, very properly. So there, he sees there is very less time, okay, I have to go very fast. Okay, so whenever the mesoporous capsules has been used, loaded with the immunomodulatory drugs or other similar type of drugs, they give better immunotherapeutic response, okay, with respect to whenever they are used as a free drugs or free medicine or as a free immunomodulator, okay, molecules with we have like immunomodulator, okay. So we have synthesized this type of, well, a lot of synthesis steps will be there. Okay, we have created this type of nanocapsules, okay, which is size 100 to 150 nanometers. Okay, we have challenged them against the macrophage. We have checked whether they can create the activity, whether they can generate the nascent oxygen or free radicals or radical oxygen or not. So we have checked with HeLa cell, with macrophage and various other cancer cells. We found that they can go and they can produce all these type of components which is responsible to kill the cancer cell. Okay, so we have checked in the layer by layer tagging of the layer by sectioning of the cells. Okay, and where they can reach also we have found. So this B image you see that is diameter from smaller means we go from higher to middle diameter, okay, and then other side of the diameter from one surface to other surface in along the direction of the D, okay, or diameter along the direction of diameter. So you see that they're carrying these nanoparticles and giving fluorescence. Okay, what we have that we have delivered the immunomodulatory drugs as a what is that? Nitric oxide. Nitric oxide is a polluting agent. Again, nitric oxide is used for the anti-cancer agent, okay? When they have a 15 to 50 ppm level are used, okay, in the particular cancer cell. Okay. is that this immunological adjuvant property has been shown by these gas loaded nanocapsules where it can be used for the hypertension, it is vascular or obstructive disease, stroke. To maintain the stroke, we can use this. Myocardial infection, we can use these things for the monitor the myocardial infection. We can monitor the inflammatory disease also. Okay, this is all are these. Okay. So function, pro-inflammatory behavior will be there, immunostimulation behavior is there, tumor suppression is there, and it can also help to cure the tumor diseases and tissue injury property. Okay, I have finished, no conclusion. So whatever the work have been done by the financial support of DST, SERB, UGC, UPE, ICMR, DBT, WELL, CSIR, and I was previously there in University of Hyderabad and presently in IIT. All, we cannot ignore their financial support or the instrumental facility they have given to us. So already 10 PhD students, 10 PhD students are completed PhD and many more are there now, 10 almost there, and postdocs who are contributing for us. Okay. Okay. I highly would like to acknowledge them. Without their contribution, we cannot achieve this type of work. Okay. We have a lot of collaborators. I cannot ignore them because under the same umbrella, under the single umbrella, we cannot get all these facilities. Throughout the world, we have this dignitaries, famous scientists are there who has helped to do various experiments at various levels. Okay. So at the end, I will be thank to the audience, and I will also thank to the organizer to invite me. And now, special thanks to the audience to listen to me. Okay, I do not know how much my voice is coming, how much it is there, audible there. Okay, so
Thank you,
this is
sir.
the end. Thank you very much. And
Thank you very
now
much for a nice
everyone
talk.
is invited if you have any question.
Yes,
out
yes.
of this talk.
Even though talk was after lunch, it was eye opener to everyone. So thank you very much for the nice talk, and we would like to have you on the campus in person in future. So thanks once again for sparing time, and topic is open for discussion if some questions are there. Any question from students, from experts? So if there is no question, I with the permission of session chair, we'll stop here. Thank you, sir. Thank you very much.
Thank you very much. I'm very sorry. I visited once your university, but this time I'm very sorry could not visit it. I hope I visit in 2015. Okay. Thanks.
Definitely. Thank you, sir.
Thanks, everyone. Okay.
Thank
So
you,
with
sir.
the permission of the chair and with others, may I leave now? I have.
Yes, sir. Yes,
Okay.
sir.
Thank you very much. Thank you. Have a nice day to you all.
Thank
Bye. 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 Take care.
So thank you one and all, and with the kind permission from the session chair, our next speaker will be Dr. M. Venkatraman. What is that? He is waiting since from the morning. So no, he requested me because he is सरांचं फेलिसिटेशन आहे का? ठीक आहे.
Yes, I would like to request Professor T.A. Kadam sir to please felicitate session chair Karyappa sir. Sir, please. Big sir. Thank you much, sir. Thank you. I would like to request again Professor T.A. Kadam, sir, to please felicitate Dr. Ved Pathak, sir. Thank you, sir. Thank you, sir. And one announcement for offline poster presentation. You have to display your posters at SLS. Offline. In tea break, you can. Thank you.
So this is my privilege to introduce Dr. Venkatramana. Dr. Venkatramana is a PhD from Venkateshwara University, Tirupati in 2001. After that, he spent almost 8 years postdocing at the different institutes. He was a postdoctoral fellow at Indian Institute of Science, Bangalore. After that, he moved to Uppsala University, Sweden for 2 years. And after that, he has also worked as a postdoctoral fellow at Thomas Jefferson University, Philadelphia, USA. And then he joined University of Hyderabad, popularly called as Central University of Hyderabad, as an assistant professor since November 2018. So he is working on the viruses basically, and he has published several papers, and 2 patents are in his credit. So first patent is on design, synthesis, and evaluation of B4N, the derivative of norhydroguaric acid as potential inhibitor of dengue virus protease. His second patent is on novel anti-NS2BNS3 Pro antibody-based indirect ELISA diagnostic test for the dengue virus infection. And he has received a grant from different funding agencies, including UGC, DBT, SCRB, MHRD, and recently he has received 65 lakh grant on role of COVID-19, NSP4 and 16 on mitochondria. So we honored to have Professor Venkatramana with us, and we'll be very happy to hear his experience with this viral research. So with the permission of chair, I would like to request Professor Venkatramana to start his presentation.
Thank you, sir, for your kind words. I like to start with for thanking 3 different occasions. First to Dr. Jore for inviting me to present here, and second, the organizers of this conference, the students and the faculty of life sciences, and the third will be for the previous speakers, which gave the opportunity to learn few more things in spite of sharing our research progress, recent progress work. Okay. So sir, please help me in. I'll try to take as less time as possible. And the title, as you have seen, please first slide. Identification of host mitochondria as the new target for dengue virus infections. As we have seen since morning, many talks, hopefully yesterday also, drug development. In order to develop the efficient or the specific drug, we should know the specific target. Correct. In order to know the target, we have to know the life cycle of the microbe. What we are is enough knowledge. The knowledge of the COVID was used based on the SARS-CoV-1 and MERS. That's why SARS-CoV-1 and MERS are not COVID-2. That's the example. So we should know the life cycle or the mechanistic insights of the any microbe to develop the efficient drug molecules. Yeah. So here of the dengue virus infections, few introductory points. Before 1970s, this disease is very much ancient, and up to 1970, you can see here. You can see in the top panel, 1970. Among the 4 serotypes across the world, only 1 or 2 serotypes were used to circulate, and there was very much limited disease severity also. After 1970, like you can see lower panel, 2004, up during which the disease become hyperendemic, means okay, light, okay. Thank you. So by 2004, all the entire globe, almost all, become the hyperendemic, means almost all 4 serotypes are circulating in each country. India is not exceptional. You can see the statistics here. You can see 1964, it is starting only 1 or 2 serotypes, but when the time goes, all 4 serotypes are circulated. At present, every state is reporting all 4 serotypes. So hyperendemic. And dengue fever is an ancient viral disease reported from more than 100 countries. Now it is nearly 130 countries worldwide, especially belong to the tropical and subtropical regions. Dengue fever is considered as fastest emerging, re-emerging viral disease, which is caused by dengue virus. As per WHO estimates, nearly 100 million dengue cases were reported every year, but recent update is nearly 400 million people are getting infection every year across the world. And the symptoms, if you see, as per the WHO guidelines, they are categorized into the without warning signs and with warning signs and the severe dengue. So the regular fever, cough, and all this comes under the simpler without warning signs. And the worry is about it is, which is like with the some of the severe complications, plasma leakage and all. You can see the rashes and the eyes damage and the skin damage, especially the liver enlargement due to the fluid accumulation and all. These are the characteristic features of the severe dengue. So what happening at present, the disease progression is, if you can see this is the during the infection, 3 to 7 days, it will be the incubation period, after which the disease expression starts and the platelet starts going down, which is at nearly 1 week, which is the hallmark diagnostic feature of the dengue virus infections. And the viremia, just before that, it will be increased, and either disease will be cured or it will go into the further complications. How the diagnosis is being carried means there is NS1 diagnostic test and IgEg and IgM based test. Both the tests are problematic because you can see here the window period for the NS1 is only very short. If you do outside this period, you will get negative only. And also the IgG, IgM, they are not this specific. Any viral fever causes it leads to the increase of IgG and IgM. So there is a problem in the dengue virus diagnosis also. Coming to the molecular virology of this virus belongs to the family Flaviviridae. Its genome size is 11 kb, positive sense RNA, enveloped virus. This is, and it is many other viruses also belong to the same family, like Zika virus and yellow fever virus, hepatitis C virus, and all. But it is more closer, similar to the chikungunya and the Zika virus. That is different aspect anyway. see the genome organization. If you see here, this is the 5' to 3' end. There will be, it is RNA 5' untranslated region, and at the 3' end, 3' untranslated region. This entire genome codes for the 7 non-structural protein 1 to 5, 1, 2A, 2B, 3, 4A, 4B, and 5, total 7, and 3 structural proteins, capsid, premembrane, and envelope proteins. Our interest is on NS3. Please remember that this is the NS3 which codes for the protease. And the current scenario in India, every year the number of cases are going on, along with the severe dengue also is being increased. For the dengue virus, almost each cell of the blood is the target cell because of the variety of receptors it is being used. It used the many receptors, so it is able to enter every cell. So that is another problem to work it out to develop the drug molecules. Not only 4 major serotypes, within each serotype, again it is categorized into different genotypes. For example, like DEN1 is differentiated into the 4 genotypes, DEN2, 5, 6 genotypes, DEN3, 3 genotypes, and DEN4, 3 genotypes. The red ones indicates the genotypes which are circulating in India. And this is little older observation. The current scenario is these genotypes are changed. Change of genotype or the serotype, it enhances the disease severity. So because of the one hypothesis, one characteristics of the dengue virus, probably in other viruses it is there, antibody dependent enhancement. Means if this season, if you get infected with the DEN1, you will have the antibodies for the DEN1 only. Consequent in coming years, if you get infected with the other serotypes, these antibodies of this serotype will not give the protection for other serotypes. Rather, it enhances the multiplication of that virus, and it leads to the more disease complications, which are raised. They are called the heterologous antibodies. They cover the virus particles of these other serotypes, and they create the environment in such a way that they multiply more aggressively and leads to the more increased So for this severe disease complications, there are many one and related to the environmental and related to the viral characteristics, and so on. Our laboratory is focused on the characterization of the viral particles as such, and what are the factors that is contributing for the severe dengue and how it is going on. So in our laboratory, we focus on many things like molecular characterization of dengue virus. Means we procure the patients with the help of the period. That is one aspect it's going on, and then analysis of host responses. This my presentation is based on this, a bit of that host response, how the host is responding for the this viral infections, and the identification of novel targets for developing among these host responses, and what are the targets in that line we can means design the drug molecules, and then identification of specific drug for dengue virus molecules, and the development of diagnostic tests. The one diagnostic kit already we have established, and it is patent is under process, and hope it will be useful for the specific diagnosis, because I have quoted the NS1 based and IgG, IgM, they have some limitations. We observed that based on this test, what we have developed, some problems we have overcome when we checked the collecting the samples, field collected samples. then we are constructing some infectious clone for the dengue virus, because every time we cannot screen the drug molecules and all, because there will be strain change, and if you use the same strain, there will be mutations acquiring and all. So to overcome that kind of problem, we have generated one infectious clone for the dengue virus serotype 1, that is in the DNA form now. So whenever we want, we make the RNA, so that RNA will be transfect the RNA into the role of dengue virus protease in disease manifestations. Means what the protease NS3 coded, one gene coded protein, it is working as the protease. It means it cleaves the entire polypeptide into different proteins, 10 proteins. They will, so that they migrate into the different compartments of the cell at the host, and they perform their activities. So in addition to that, in addition to the specific helping characteristic for the virus itself, it is damaging the host also. There are some substrates we also identified, some other labs also identified, due to which host is getting damaged. So this virus, this protease is a serine endoprotease, and it has kind of 84% to 3 and 83% to the 4. Means the rest 67 means 33% difference is there between 1 and 2. So it is the differences between the serotypes also. So the basis for our present work is, some time back we collect the samples for RNA isolation, and also that same serum samples we have analyzed the by the STS page analysis. What we have observed is, some of the clinical samples, there is some proteins disappearing on the top and some proteins appearing in the clinical samples. And for these samples, since they are doublets, we have run the gradient gel and separated enough that two bands, and eluted this band and this band, eluted and sent for the mass spec analysis. We found that this band is EDRF1, erythroid differentiation factor 1, and these two doublets are alpha spectrin and beta spectrin. Then see, we worked some like literature survey. There we found that this real molecular weight of this EDRF1 was 135 kilodaltons. Then why it is migrating so low? Then we checked whether any cleavage site is there for that protease, dengue virus protease. We found that protease cleavage site over here. This end is the 3' C-terminal end of this big protein. So there it's started. Then further literature analysis based on that, we made some hypothesis. In the normal tissue, the EDRF1 will be needed to activation of the GATA1. GATA1 in turn binds to the upstream region of the spectrin mRNAs, and that spectrin will be synthesized more, and this spectrin is required for the platelet formation. So because in the infection condition, the EDRF1 is being cleaved, so there is no activity on GATA. There is no activity means there is no spectrin. There is no spectrin means there is no platelets. This is the thrombocytopenia is happening in case of dengue virus infection. This was our hypothesis few years back. Based on this hypothesis, we have constructed some of the clones. Like this is the RNA, for example. These are the proteins, different proteins. Here is the NS3, and the NS3, if we elongate this, the NS3 itself contains protease and helicase, and this is the protease without NS2B. This is the red portion. This is the red portion. This red portion, it indicates this MTS, mitochondrial transport signal sequence. So it indicates that this is going into this may go into the mitochondria. So to test our hypothesis, we have collected some cell cultures and transfected the cell lines with the only EGFP vector and the different constructs. Means in this experiment, if you get only GFP, that means it is only vector GFP. And if you get the mito RFP along with NS3 GFP, we have also transfected the mito RFP. Means if RFP is there like this, that indicates the mitochondria. They are mitochondria. Wherever both are there in this kind of mixture, that means this protein is reaching to the mitochondria also. So our hypothesis confirmed that this NS3 GFP, it is reaching to the mitochondria along with the mito RFP. So then along with our one of the collaborator, we have performed the in vitro assay to show that this protein is indeed entering into the mitochondrial matrix. In the history of this animal viruses, as per our knowledge, a virus coded protein entering inside the matrix is the first report, first observation, because there are some other viral coded proteins that enter, that reaches up to the mitochondrial membranes, not the matrix. So okay, based on that, what it is doing entering into mitochondrial matrix. So whatever the antibodies we have in our laboratories, we have checked some of the proteins, their levels, like the GRIP1, GRIP-EL2, ASPOS, and all these things. Among these proteins, one protein called the GRIP-EL1 showed the some level decrease in case of transfected conditions. So then what happens? We made some mutants in the catalytic thread. We transfected that also. That is not cleaving. There the GRIP-EL1 level is not going down. Only active protein only it is doing that decrease of the GRIP-EL1. This GRIP-EL1 protein is the co-chaperone along with GRIP-EL2, which is helping the HSP70, mitochondrial HSP70 inside the mitochondrial matrix. Mitochondria, you know, we all know that it is powerhouse of the cell. It is required to generate the ATP along with proper folding of the proteins, many other functions, many diseases also it is involved, mitochondria. So, so as a first step, we identified one substrate called the GRIP-EL1 inside the mitochondria. Then we have, since this protease is the artificial construct, we have generated the list of constructs where I have shown. The natural construct also means NS3 helicase. That itself we have transfected where the GRIP-EL1 completely disappeared. We have infected the cells and see the GRIP-EL1 less. It is very much down compared to the construct itself. In natural, what I mean to say is in natural conditions, more cleavage is happening than the in vitro studies. So both the NS2B, both the NS3 protease and the GRIP-EL1 proteins, we have cloned into the prokaryotic expression vectors and purified them separately and incubated in vitro. So you can see the cleavage of the upper band and increase of the lower two bands. Cleavage is going on, Western blotting this is. And then along with GRIP-EL1, GRIP-EL2, we have procured, means expressed and purified, which is not being damaged anymore. And then we checked whether the cleavage site is there in the GRIP-EL1 or not. Yes, it is there. Two sites are there. Two sites we have mutated. In the mutant versions, this is not affected. In the mutants, both the versions, it is not affected. So in this way, we have confirmed the specificity of this NS3 protein, the cleavage of the GRIP-EL1. Okay, there was one question that if why not these sites are there in the GRIP-EL2 also, but it is not being. Then we superimposed the structures of both. Only in case of GRIP-EL1, the cleavage sites are coming closer to the cleaved, and it is only being cleaved. So what is the scenario of this in case of clinical samples? If this is all entire thing is true, it should happen in the patient samples also. We collected many hundreds of samples here. I showed here only those gel, and which controls DF, DHF, and some of them completely going down, GRPL1 levels. And corresponding platelet counts, if you see that, wherever completely GRPL1 is going on, that patient's platelet count. So that means the understand that what it is doing in case of normal platelet count ultimately it's not working. Yeah, yeah. In the normal case, the cells import the proteins to the nucleus and to the mitochondria and all, and EDRF1 binds all right to the GATA, and GATA binds to the promoter of the spectrins, and spectrins synthesize enough, and as a result, enough number of platelets are being synthesized. Whereas in case of dengue virus infections, this infection leads to the cleavage of the EDRF1, and there are two forms of the protease, one form entering the mitochondria and cleaving the GRPL1, the second form entering into the nucleus and cleaving the EDRF thrombocytopenia mechanism in the dengue virus infections. Okay, so in this line, we also gave an attempt to identify some kind of drug molecules targeting this protease only. So for that purpose, we have expressed the NS2B, NS3, which is the complete protease, and purified, confirmed by Western blotting. Then its activity, we have procured one fluorogenic compound with which, if you incubate this protease at pH 7, which is we established this one also, and it's the fluorescence will be keep increasing. Fluorescent as the concentration of the protease or the substrate or the enzyme increases, fluorescence will be keep increasing. Under this background, and the molecules we have procured with the some collaborator from Bangalore Institute, Ramaya College of Institute. What the background of this molecules is, there is a plant product called the NDGA, which is proved that it inhibits the dengue viral infections. Not only dengue viral infections, many other viral infections also it's being used along with the quercetin, another molecule. But both the molecules, the problem is they are like highly cell toxic, so they are not come up to the public use. So here hypolipidemic agent with the antioxidant and anti-inflammatory properties inhibits the formation of virus replication complexes. Cytotoxicity of NDGA was identified to be high, so it was not used. And so what we generated two compounds from this by making some side chain changes, and this is the mechanism he has used to make some changes here and develop the two molecules called the T4N and N and B4N. So their cytotoxicity, what we have measured, like MTT assay, somebody was talking in the previous talks, and the cell toxicity, if you see, for example, in case of quercetin, NDGA, these two are lesser than these two. Means to kill the cells, it is required more concentration than compared to these two. So in that way, first we have checked its cell toxicity is less, and took forward. We have checked their binding ability using the CD spectral analysis. As we increase the concentrations, the binding is going more and more. We calculated the KD values, and finally we checked its inhibitory concentrations also, inhibitory concentrations ability also. So the final goal is to protect the mitochondria. That is being protected or not. So that is being there. See, in case of NH2B, NH3 transfectate, the mitochondria is being discreted, the damage, which is protected with this both the molecules. So then we compared with whether it's qualification as the drug molecules using the CC50 and IC50. The selective index is higher than these control molecules, especially in case of T4N, it is much higher. So we are thinking that this drug molecule can be taken forward for further analysis. So our thanks are due to many funding agencies, without which the work will not be possible. And the students who are involved, the work presented here is done by the Lekha and Chaitanya. Thank you, Ananda.
So thank you very much, sir, for the nice talk. It is very important because we all have faced COVID and we are still facing the fear. So you have, and your efforts have already provided some for this antiviral therapy. Topic is open for the discussion, and we can take one or two questions, if any. Questions? So if there is no question, we would. Yeah. Okay. So how these proteases from dengue and can be used as a model for targeting COVID? So
thank you, sir. That line of work also is under progress. There are two proteases in case of COVID. Both we have like similar story, only it's going on. It's incomplete. So that's why I did not bring it here. Okay, thank you. More susceptible possibility is there because the immune system already will be in compromised state, I feel.
Is there any way to use some means because as we know, COVID is inducing this immune response, right? A cytokine storm, right? So can we use some of these leads you are developing for inhibiting those cytokine storm or immune response or hyperimmune response per se?
We are not yet at that stage at this moment.
Yeah, please go ahead.
Yeah, it will be more. No, in the first instant itself, the viral load will be more. It will be more dangerous only in the first stage itself. Because in our experience, we had the sample from one child. We brought the samples to the laboratory. We identified all 4 serotypes at a time, and by the time we gave the result to them, she is passed away, unfortunately. Like,
So with this, I would like to request Director Professor Chauhan sir to felicitate Venkatramana sir. Thank you, sir. So with this, we would like to proceed for the next session. Next speaker is Professor Radhya, Professor Moria from University of Hyderabad, and he has completed his master's from Banaras Hindu University and PhD from Banaras Hindu University. He has been working in University of Hyderabad since from last 13 years, and he has published several papers of in the journals international repute. He has awarded with several national and international fellowship, like Raman Fellowship, where he worked in for his postdoctoral research in USA. After that, he was also awarded with ICMR DHR fellowship, long-term fellowship for postdoctoral research in NIH, USA. His area of research interest is in mechanisms of infections and immunity in visceral leishmaniasis, identification of new drug targets and drug-resistant markers to control leishmaniasis. So with this, we feel privileged, sir, for your presence, and we will be very happy to listen your experiences with leishmania and your experiences in NIH and US FDA, where you have completed your postdoctoral research. Professor Moria, sir.
Thank you, Dr. Jore. So I have the touch with Dr. Jore last 4 or 5 years, I think. And he keep insisting when this event is organized by the School of Life Sciences. So I also thanks to the director, School of Life Sciences, faculty and students sitting here and giving this privilege to speak here, whatever we are doing back to the University of Hyderabad last 13 years. So me and Venkatramana both are got position in the same interview panel. And he was in the biotechnology, I was in animal biology. And in before interview, we met and we have the some share experience. And after that, he got by that time, because I am from Banaras, he is from Tirupati. Many of the colleagues are from Tirupati, they inform you are selected. So once I reach after I think when I came to join, then because we have such a simple conversation before interview. Then we are since working in the my School of Life Sciences together. He work on virology, I work on the parasites biology, and I did just like what he said. I work on the leishmania. So I will share what I am going to do urgent here. Hope I will also take short time, not and finish the within the your what you assume. Okay. So what title is there? Bifenthrin, a Bithynia somnifera, a potential drug to treat the visceral leishmaniasis. Actually, I work on the three perspective of the leishmaniasis. One is the drug marker. Second is new drugs, the parasites, how escape from the host cell immunity and survive inside the host cells. There are three perspectives, but going to talk only one perspective, identifying the new drug target or new drugs. Yeah. So before going to the leishmaniasis, I may assume everyone know, but just I will brief what is the leishmaniasis. Leishmaniasis is a vector-borne disease caused by the protozoan parasite called Leishmania, and it's spread by the mosquito. Basically, it is the disease is very common in the poor society of the very high humid areas. So the disease is spread across the world, nearly all continents, and have the 88 country, and majorly is the having the major country affected is the India, Bangladesh, Sudan, Brazil, and some Latin American countries. So most of the cases come, and but what the and the disease have the three different kind of the manifestations is cutaneous, mucocutaneous, and visceral leishmaniasis. So I will go by one. So if you look at the parasite, when sandflies bite to the so when sand okay, so when sandfly bite to the human, it transmits the parasite to the human, and it goes generally in the human. It like malaria life cycle that it goes to the RBC, but here it goes to the immune cells. That is called the first defender is the macrophage, and we know the macrophage is the having the ability to present the antigens to the T cells to maintain the Th1 type of immune response to protect the protect the host from the any infections. But this parasite target to the macrophage, and it multiply inside the macrophage. So another challenge is that this is the intracellular pathogens. So it is targeting the intracellular pathogens is very tricky because your parasite sitting inside the host cells, and recognizing the target of the, if you identify the molecules, is the very first task is that the drug is should be penetrate in the host cells, then only can it reach to the parasite. So these are the all intracellular pathogens still is the challengeable, like Mycobacterium tuberculosis, tularemia, and leishmaniasis, and there are several other intracellular pathogens. So they are still in the challenge in the across the world. Even we have the very much advanced the science in the infection biology, because they sit inside the host cells. So the life cycle pass through this, and again, when sandfly bite, it goes to the sandfly where it survive in the gut, and the life cycle keep continue. So if you look at this disease, what I told is mostly the India, Brazil, and Sudan have the major 90% cases of the visceral leishmaniasis. The 18 country of the Western Europe's, the developed country, they also affected with this disease. So a lot of the investigation is going even not in India, across the world. Can it change? Different kind of the manifestation again extended 2025. Is more or less is eradicated, but still is the some sporadic area of this endemic region still is this disease appearing. So what is the important is that, can you change? So what is the pathogenesis of the BL, which is the Indian perspectives? So what is the pathogenesis? BL is the life-threatening and having the Th2 type of immune response. I don't know how much in the student group they know the whatever the Th1 and Th2 type of immune response. Just I can brief. Th1 type of immune response protect the host immunity against the infections. Th2 is the help to pathogen get proliferate inside the host cells. So Leishmania once enter into the cells, they direct the macrophage in the Th2 type of immunity. So they have make the macrophage in such a way that they can multiply and they grow, proliferate faster there. So this and what they lead to the make Th2 type of immunity is that they have increased the IL-10, TGF beta and IL-4 cytokines, which are the very important to make the means, you know, one word is used that exhausted T cells, means the cells are there but they are not functional. So these cytokines, they make those cells, even if you look the microscopic, they are very normal, but they don't respond against the pathogens because internal signaling is the very much controlled by the parasite. Then what is the important is there, all these lead to impair the acquired immunity against the VL. So once, if you left untreated, that's what in the beginning I told, if you left untreated, the 100% fatal. Person cannot survive longer. Please next. So there is a very limited drugs are available to treat, and there is a, if you look there, sodium isotretinoin, sodium amphotericin B, miltefosine, and pentabins. These are the 4 drugs, but all that drugs are very toxic. And this anti-isotretinoin band is not in use because there's a lot of the resistance against that. And amphotericin B is still in use, but it is the, you know, as amphotericin is a nephrotoxic and it is required to hospitalization to give this medications. And miltefosine is the very recent, we can say like 10-15 years back is introduced by the very privileged, my supervisor is a Professor Syam Tunzar in the Banaras Hindu University. He introduced this medicine and it is the first oral drugs and to treat the visceral leishmaniasis. Rest are the all is the intravenous and need hospitalization at least minimum 2 weeks. Okay, so there is no vaccine. There is no vaccine. So what is the important? Only you have the only one oral medicine. Now it is after 15 years of the use, this miltefosine in the endemic area, they are showing is more than 20%, again is the resistant problem. So it is this parasite, as we know, like we are talking about the COVID, this 1 version, 2 version, 3 version. So the pathogen have the mechanism to evolve to against the resistance. So you have to keep working on and identifying the drugs, new compound. So based on that, we are working on the natural products, and where we have going through the review literatures, and we found that betaferin have the probable anti-protozoal activity. There is a reported is anti-cancerous, anti-inflammatory, antioxidant, and antiviral. Is a lot of the, and even in before COVID, they say you have to use this betaferin, you boost your immunity. So we also back to the 2012, we started working on that, and we identified in there one compound is the betaferin A, have the potent anti-leishmanial drug. So how we lead, and I will show in the further slides. So first, what we have used the crude extract from the leaf, and we tested as a anti-leishmanial activity, and you see that it is it's, yeah, what is you changing? Can you make it? It is going somewhere. Just back. No, you are going ahead. Just like, yeah, just wait. So we treat this crude extract. We have the several fractions, but what I am here is there is a fraction 4 and fraction 5 and 6, and we found that is so the very potent anti-leishmanian activity. If you see the IC50 52 microgram per ml, and another component is have the 13 microgram per ml. Then we have done the all LCMS and other things. Then we identify the what are the compounds are there, and we further tested this compound. These are the showing the electron time. And there, if you see that 24 hours treatment and 48 hours treatment and 72 hours, the parasite getting degraded, their morphology is changing. And then we tested by the tunnel assay, is called as anti-apoptotic markers, and we testing, and then we found that it is showing the early apoptotic kind of the death when we are treating with these fractions. Yeah, next. Then we further see when we are doing the any anti-leishmanian compound or anti-inhibitory molecules, we should not just only have the IC50 or some of the 1R experiment. You have to multi-dimensional test to your compound is working properly. So we look the tunnel to see the DNA degradation. Then we have done the look that's DNA cell cycle analysis. And if you look at 24 hours, it is giving the that in this compound is inhibiting the cell cycle, arresting the cell cycle in G1 GFH. Then we further go into the see that what is the early apoptotic markers. So if you look that is this is annexin V staining, and it's so here if this one is the miltefosine, this fractions. If you see miltefosine, which also have the apoptotic like death. So if you see the more death is happening in the miltefosine, same we found in the this fraction 5 and fraction 6. Then we further want to say that want to check that how it is the damaging the cells when treating with this. So we look that the mitochondrial membrane potential. This drug is entering into the mitochondria and reduce the ATP generation. So cells are compromising to the further multiplication. So we tested in the mitochondrial membrane potential, and you can see that membrane potential will getting decreased. So means if membrane potential get decreased, parasite can't produce the ATP, and then it becomes a low energy, and it further cannot multiply. Then further we tested to the again is the looking in the what is the reactive oxygen species. One of the important factor when you are targeting the any pathogens, you have the 2-3 place. One is the mitochondria, second is the DNA degradation, and third one is the membrane. But here what we are looking, we saw that the DNA degradation is happening, mitochondrial membrane potential also changing. If mitochondrial membrane potential will change, it changed the reactive oxygen species, because reactive oxygen species is the byproduct of the electron transport system. If you have no electron transport system and the complex A3, when the last step of the A3, when its formation of the ATP happen, if it's the block that step, that reactive oxygen is innocent oxygen will be available that can further damage the particular organism, particular cells. So we have the several way we try to prove that this compound have the apoptotic like effect. So what we conclude that morphological alteration, DNA fragmentation, arrest of the G1, G2 phase, G0, G1 phase, and externalization of the phosphatidylserine serine, which lead to the membrane potential damage. So this paper was later on published, and then we move to the, once we understand the in vitro model, then we go for the ex vivo model, because this parasite sitting inside the macrophage. So what is happening in there? So please next. So what we do, before once you treat the parasite directly with that compound, you have seen that is apoptotic like that. But when the parasite sitting in the inside the host cells, how you have to treat, how you decide the concentration of drug, you have to first test on the naive host cells. So we tested the cytotoxic effect of the macrophage, and we looked that there is up to the 25 microgram of this 80% cells are survival. Means this is the not damaging the host cells. Okay, are you getting point? But And if you look the same other in that another compound which is the present in the fraction is the beta-ferrin A with the major fractions, we also tested that one, and we found there nearly, if you look at the 5 micromole, it is nearly 80% survival. Means this compound is not damaging the host cells. So that may be easy to take the next level. So what we did, we have infected this macrophage and then treated took the this fractions is a concentration of the 15 microgram and 10 microgram. Because it is the up to 25 microgram, there is no toxicity on the host cells, on the human cells. So we took this concentration and we treated that infected macrophage, and we found that they are drastically reducing the parasite load in the infected macrophage. And then you can see that this, and then when I just before I told this parasite is this compound induced the involved in the electron transport system, membrane polarizations, and control the reactive oxygen species. So we want to look that what about the nitric oxide, how it is nitric oxide, because macrophage have the two way of the mechanism, or it can kill the parasite by the reactive oxygen species or by the nitric oxide. So these are the super oxides, as we know. So they are the very much potent to bind to the membrane of the cells and then damage the membrane potential. So we found that this fraction 6 is, if you look that nitric oxide is getting there, but LPS is getting up. Then we have now nitric oxide should increase, but it decreased. So what is the reason if nitric oxide is getting decreased, how the parasite getting killed? Then if you look at that another here, this perspective in the same beta-ferrin also behave like that. So we test the further experiments. Just please next. So if you look here, so what is this is the reactive oxygen species. I told there is a 2 of the superoxide is required to clear the pathogen inside the host cells. So we looked at and we found that reactive oxygen species getting upregulated either nitric oxide. So macrophages are able to induce the reactive oxygen species which can clear the parasite infected inside the parasitophorus vacuole of the macrophage. Then we also test whatever the macrophage. Macrophage, we know they produce the several different kind of the pro-inflammatory cytokines. So we check the pro-inflammatory cytokines, IFN gamma, IL-10, and then we tested the ratio. So if you look both, IFN gamma is the pan marker for the any kind of the superoxide generation in the cells. But here is the silent. Then my result is contradicting. Other one side we are seeing the superoxide is ROS is getting increased, but IFN gamma is not silent. So where is the link? What is your data? But important is IL-10. IL-10 is drastically decreased. And I told in the beginning of the pathogenesis of BL, TH2 cytokine, IL-10 is the helping to this parasite survive better in the during the infections. So what is it doesn't affect the IFN gamma, but it affects the IL-10. Means is a reverse way it is working as a to induce the superoxide. Okay, so ISD. So that way, so once we look that IFN gamma and IL-10 ratio, you can see that fraction 5, fraction 6, and everywhere is getting upregulated compared to the control. So then we prove that this IFN gamma is not IL-10 getting down-regulated. IFN gamma even is the constant, it having the positive response and inducing the superoxide in the macrophage. Then we thought, okay, next we have demonstrated in the in vitro direct parasite, then ex vivo. Now we thought, okay, let's go in the mice model where it is what is happening in there. Next, please, next. Just next in. So when we what we did, we infected the mice with the parasites, and we treated with this fraction and this beta-ferrin alone. And if you look that this is spleen, because this parasite visceral leishmaniasis is having the enlarged splenomegaly and hepatomegaly. So we saw that what is happening when we are treating this compound, it is drastically decreasing, significantly decreasing the parasite burden in the spleen of the mice. And even in the when we are using the pure compound beta-ferrin A. So what is the and then we result we found in the liver and liver in the in case of the fraction, in case of the pure compound. So what is the goal? We have this compound is what is doing. One way is doing directly attacking on the parasite, making their membrane potential of the mitochondrial membrane potential of the parasite to damage. So the parasite cannot generate more ATP and it cannot survive. Other way, it is inducing the host cells by the inducing the positive immune response to clear. So you both result what in vitro we have shown, it we have seen directly in the mice model. Next, please. So we also see that demonstrate the liver sections, and we see that granuloma formations in the liver. This is the control, and you have the very big. You can say that's very clear granuloma formation is there. But when it is treated, you see the very small that is reducing the granuloma formation. Having the both way of the one is the granuloma. You know granuloma. Granuloma is the formation in the liver when liver macrophage protect make the rosette formation where the infectious parasite is there, and make in such a way that there is a more less oxygen supply, less nutrient supply. So parasite cannot propagate more. If your liver granuloma is healthy, it will suppress, clear the parasite. But if is the pathogen are stronger, then its granuloma formation is become bigger, and you have the more parasite like what we are seeing in the control, untreated. Please next. Then we want to see the another cytokines, because in the when we have seen the parasite load is decreasing, granuloma formation is decreasing. What about the cytokine response? So if you look that cytokine response the same, IFN gamma is not responding. There is no significant. IL-12 is also we are not getting is another pro-inflammatory cytokine released by the macrophages. And INOS also, because NO we have not seen any significant, just the ex vivo model also. But if you look the Th2 cytokines, what is the happening? There is significantly decreasing. So what is the meaning? This beta-ferrin is inducing, suppressing your Th2 dichotomy of the immunity. So it is suppressing where is the if you have the Th2 type of dichotomy, you are very much susceptible for the infections. So it is controlling the cytokine which is involved to weaken your immune system. So they suppress those cytokines. So your immune system will be always will be strong. So if you look the IL-10, TGF beta, every is getting shut down in the treated cases. Please next. And then if you look in the same is the that is the fraction. This is the purified beta-ferrin A. And please next. So what I told in the ex vivo, same ratio we found in the in vivo model of the mice, and we measure this IFN gamma and IL-10 ratio. We found that is the whenever you have the beta-ferrin A treated is a very good. You have the miltefosine is a control treatment drug, and you see that is we shoot up the IL-10 IFN gamma IL-10 ratio, and same like in the fractions. Please next. So please next. So what is the now we have started from the parasite ex vivo and in vivo, but still question is there. What is the exact target? In the morning sessions, sir told we are not getting the patent, but we are not realizing why we are not getting the patent. The reason is we are not demonstrating as per the FDA norms. FDA, you must everyone know, Food and Drug Administration of the USA. They are very, very extreme, very rigorous, review the whole your experimental. Then only they approve the drugs. So a lot of the things we cannot just simply crude extract, we treat and we have this thing, we have the turmeric, we have the antioxidant. No, you have to have demonstrated the mechanism inside that. Unless mechanism, nobody will give you. They say तो कोई भी कर सकता है, कहीं भी दे दो वो grind करके दे देगा और कुछ ना कुछ कम हो जाएगा. Even if you take the alcohol, 100% alcohol, put in the culture, all will cell die. So can I get the patent on that? No. You have to have so the mechanism. So we thought let's go inside what is the actually happening this compound, how it is working. So if you look this folate biosynthesis pathway, it is the well known in the target for the any infectious disease, any kind of the bacterial or protozoans infections. Always they target, try to look that folate biosynthesis. Like we know, whenever your doctor recommend the antibiotic, they say you take vitamin B complex. So why antibiotics? Because that pathogens in the gut who synthesize folate for you, they will die. So you have the sort of the folate. So you need a supplement. So we also look in the parasite. What is happening in the folate biosynthesis in the parasite? Is a big parasite have the 36 chromosomes. We have 46. So you can imagine as a single cell parasite. So how is the complex? 36 chromosomes. So it's a very complex. So you cannot go each and every. But whatever the established model and the review systems in literatures, we check first, okay, let's see in the folate biosynthesis. What you're seeing here, this is the human host path, human, this folate biosynthesis happen. This is the parasite. What is the important here in B, where is highlighted? Parasite, human have the dihydrofolate, but parasite have the two systems, dihydrofolate and pteridine reductase. If you shut down this enzyme, parasite can use this and it can make the folate and it can survive. If you shut down this, parasite still will survive because they have the another enzyme. So they have the alternative. So that is called in the parasite biology who are teaching your, they must have talked about the longevity. One word which came in the longevity of the organisms. How they have their two host digenetic life cycle. Without they cannot survive, but still since ancient they are still existing because they have the different kind of the strategy to survive. So we think in that, and then we thought, okay, we start to analyze this data. Where is it our molecules is interacting? So we enter, please next. Please just go next. So what we do, we take the pteridine reductase first. We dock it with the beta-ferrin A, and we saw that is a binding with the that interacting with the pteridine reductase enzymes. Then we did the enzyme assay, enzyme activity is the 6 hours and 12 hours and 24 hours. You have the control and you have the beta-ferrin. And if you see that beta-ferrin is significantly is decreasing this activity of the pteridine. Then what is the interaction is there? There is a, if you look the docking energy is 6.73, is considered as a 5 is considered goes to the 10, 25, but not more than much energy is there, then irreversible. It cannot, then is problem with the excretory. In your body will not excrete. It will be intact in your body. So that is also drug discovery. That is too important. Drugs should be irreversible. It should be come out from your body. It should not hang over there. So we found that it is inhibiting to the pteridine reductase. Next, please. Then in silico study, then we thought, okay, let's clone this protein and let's go in the in vivo models. So we clone, express, and then do the enzyme assay. And if you see, just go next slides, please. Then we do the enzyme assay using this pteridine reductase enzymes. And you see that when is the concentration optimized, we have the 0.69 micromole is the optimum concentration of the protein. And we have using the bioterrin and it's following the Michaelis-Menten equation. When we use the inhibitors, what is this graph is saying? You must have the enzyme kinetics. You have studied this graph, Line-Weaver-Burke plot. What graph is showing? Anyone, any student can say what graph is showing? This graph is showing first Vmax also changing and Km also changing. So when Vmax and Km both change, what kind of the inhibitions? Is uncompetitive inhibitions. So this drug is working as an uncompetitive inhibiting the pteridine reductase. And that's very important when drug discovery we do. we don't want competitive inhibitors. Because these competitive inhibitors, your other enzyme, your host enzyme also get inhibited. So you need uncompetitive which can bind to the other, then inhibit your particular target enzymes, not to the host cells. So that is the considered is an uncompetitive inhibitor. Most of the drug is the uncompetitive. There is a non-competitive also, but most of the drug non-competitive is a competitive inhibitor. Generally in the poisoning case they use. When you have the alcohol poisoning, you have to ethylene glycol. That's the competitive inhibitors. So in that way, you have to have the drug which work as an uncompetitive way. Please next. I'm just finishing. Then once we prove that PTR1 is inhibited by that beta-ferrin A, then I told there is another enzyme is also the DHFR, dihydrofolate. So we again went for the docking and checked that how it is this. Next, please, next. So we dock that. This is the DHFR-TS. This is the is a domain kind of the protein and having the two independent domain. This is DHFR and TS. And you when we dock this, this is the red is showing the substrate DHFA. We have the beta-ferrin A. So you see the substrate is binding into the matrix is the competitive inhibitors. That's what when you are taking the antibiotic, in the other case, we have to take the vitamin stuff for that supplement your folate. But in this case, beta-ferrin is not binding into the active site. It's not competitive. It is binding somewhere is different. That is called a channel of these two domains because both are the independently work. Both there is a name is the DHFR-TS, dihydrofolate, and thymidine synthetic. There's two combined the bifunctional enzymes in the parasite. But in human, both are the totally independent. And they are located. If we dock completely, then you see that substrate, your inhibitor, non-inhibitor, and then we have the beta-ferrin A. And what is the energy, binding energy? If you see, DHFA substrate is 8.8, MTX is 8.9, and beta-ferrin is 10. So have the better affinity to bind to your dihydrofolate TS. Then please next. And now you look that human. Then we also check the human. What is happening in the human? This both protein is located in different locus in the chromosome. They are totally independent. But in Leishmania, both are the together and act as a bifunctional. Here, you see all they are. This all a question is again it come. It is binding to the humans. Human hydrofolate also. Then it is not a good drug target. Is it? But if you look the affinity, the parasite affinity have the more than humans. So it can be used. Many drugs is in the system, which if the affinity have the target pathogen is higher, they can use for the treatment. That's what is have some list. Okay. Please next. So we do we have done human DHFR, the human TS, and you have the DHFR. See 131, and more or less is the near to the twofold, 1.5 fold is higher than DHFR TS of the parasite compared to the human. So it has the very good target. These molecules can inhibit the DHFR TS2. So you have it can inhibit the PTR1, it can inhibit the DHFR TS2. So both parasite, both target can be inhibited, and then it can control. So we conclude that due to the please next, due to this region. So we also clone these enzymes, and we have also tested the enzyme. Please next go. And if this in general, we have done this. In general, is this DHFR TS use the NADPH to convert, and this NADPH we measure as a fluorescent intensity. So if you have the DHFA as a substrate, and you see the OD at this near to the 1. But when you are coming to the MTX inhibitors and vithaperin A, MTX 0.4 and vithaperin A also start from, but drastically shut down this enzyme activity. So this conversion of the NADP to NADPH is not happening, and that's what is the OD you are getting very less. So what we conclude, please next slide. I will not talk our top one. I will talk this one because top one is always we have ancient history. Ashwagandha have the lot of the immune booster. I will talk here. What is there? It inhibits the both enzymes PTR1 and DHFRTS on the uncompetitive modes. So that may be the reason when we are started from the in vitro to the in vivo, we are seeing the drastic reduction of the parasite load in the animal models. So it is and we have filed for the patent. I told in the beginning, it's not like that you have the some information, then you get the patent. Even if you put there is a lot of the other information. They have asked several other information, like they are asking, you have to have show the drug and protein ligand binding through the SPR. One SPR is the method, CD spectra. So many other experiments they have asked. We have to do, then I can get the patent. So it's not like that just you have say this is the having this property, we are not getting, they are getting. Second things also, Indian government are very stringent. If the compound, any plant is patent for the X, you cannot get the patent. But US system is a different. US system, even they give the patent on your methodology also. If you make the new methodology, method, that also. But Indian, if the drug is discovered for the diabetes and it discovered for the cancer and patented for the diabetes, you cannot patent for cancer. So Indian patent law also is the very stringent. So these are the lacunae in the system. So sometimes people doesn't know. So say we have. So these are the problems getting the patent and these things. We can file, we can get the US patent, but the Indian not, because it is already patented in Indian system itself. So they say once is patented, you cannot. And he's a very big scientist. I think he's a Maharashtra from the Rajiv Gandhi Health Sciences in Pune, Professor Patwardhan. He and me filed together. And I started 2014, and one senior professor in the order, he's a very much industrial person. He said, why you are not filing? I said, sir, let them PhD do. I don't want to have any business. When one of my friend is the patent attorney, when I used to explain that you file it, why you are waiting? When he starts searching, then he said, already just 1 year back, it is patented. Then I saw that Professor Patwar and all detail came like that. Okay, thank you. Too nice to hear me. Yeah.
So thank you, sir. It was a very nice talk. Very good data, very nicely presented, and within time. So that need to be appreciated. Please have a big round of applause for him. And a very nice piece of work he has presented. And then it was very apt for our theme, that is advances in bioactive molecule. So he has shown how exactly we can start with a plant, and then we can directly lead to identification of a bioactive molecule through its understanding its mechanistic this thing as well. So thank you, sir. Thank you very much. And we also basically these immunomodulatory molecules are very interesting. We also proposed one proposal on TB, that immunomodulation. Karat sir was there. We both were presenting before DBT task force. Unfortunately, I could not get that. But yes, this is very interesting area, immunomodulation. And thank you, sir, for enlightening us with your this excellent presentation. So topic is open for discussion, and we will expect some questions from the student audience. Maybe 1 or 2 questions. Yeah. Yes. Yeah.
Since, yes. Yeah. Yeah. No, no, it's not upregulated. TH2 is the response down. Yeah. See, in case of the parasite, we saw the mitochondrial membrane potential change. We have not talked about the host cells. In host cells, we are talking because this parasite modulate the TH2 type of immune response. It induced, so what happening when the TH2 type of immune response like IL-4 and IL-10, it is increased, macrophages are look like healthy, but are incompetent to clear the parasite via nitric oxide production or superoxide reactive oxygen species production. So they control. This is the very, and we have the one model of the, in 2007, we published a very excellent paper in, when I'm doing my PhD. When we took the splenocyte from the patient and we blocked the IL-10 using the anti-IL-10 in the body. And once we block and we put the culture, the drastic parasite growth is there. So what happened? Sorry, they reduce the parasite burden. Means once IL-10 you block, the other side of the TH1-2 dichotomy is get TH1 dichotomy get upregulated. Means IFN gamma it come in functional. So it's a kind of the system is the a kind of the reflectiveness. Cells are there, everything is there, but they are the lost somewhere in the signal. So they produce the signal like TH1. So they cannot induce the nitric oxide. Basically, it's nitric oxide produced in the mice, and reactive oxygen ROS is produced in the human model. There is a difference. So what I took here, there is a mice cells. That's what we are looking the nitric oxide, but nitric oxide is level is silent, as I showed that. But at the same time, I am saying that ROS is getting increased. So that is there. So that part is the why I not stress too much, because already we know, we say Ganda is the immune booster. Uska naam hi os rakha gaya isliye ki you eat and then you have the घोड़े जैसी ताकत आपके अंदर होगी. तो कहने का मतलब इसलिए उसका रखा नाम ही रखा गया. It had the inducer of the immune response. But my goal is not that one. I want to show because it is already established in the infection model. My goal is that once the infection is happening, how it is that one dichotomy is in the immune response, other dichotomy, other perspective is that is parasite directly killing or not. In parasite is in target is there or not. So that's what we enter into the looking the parasite followed by cells.
If you have an extract and getting to compound, HPLC
No, actually, that's what generally I have my ability to keep like that 20 slack and 25. Because sometimes people get lost and they don't ask any questions. That's what I keep narrow. I make the somewhere they have the some question. There we have once we this fraction, we have the several fractions we tested. And their fractions only 5 and 8 and 6 we found there is a having activity. Then we go and we see then there is 6 or 7 compounded with a methylite A, methylite G, and methylite H. You know, with how many methylite is as of now discovered? More than 1,000. More than 1,000 methylite. And
so many
yeah,
how you figure out
so yeah, so once we fraction, we look this fraction 5 and 6. We can go for the LCMS and we found that one fraction is 7, another is the 8 or something. Among them, again is the problem. Again we narrow down how? Again we narrow down because all are the not commercially available. This work is the 2013 or 14 somewhere. So beta variant is available, and beta variant also is present in that fraction. So we thought IJFC
as a research scholar always think, sir ये है इसको पहले करते हैं जो नहीं है उसको बाद में देखेंगे. So आज भी we are still working on that to fractionate the all compound that is not available. Market is new molecules are available and it is very costly. 50,000 you have to pay the molecule for 5 kg of this. So we are still working. The next level we are working in the other ratio. So we narrow down that LCMS data. There we see the vitamin age there. Okay, let's check with vitamin. Then we it. It may be other compound, whatever the 6-7 fraction is there, may be more potent, but that is not available. So we have not done that. So we are working. We have closed that.
Thank you, sir. Any question? So with if there is no more question, I would like to take this privilege to felicitate Professor Moria sir on behalf of organizing committee. Please, sir.
Thank you, sir. Thank you, Zora, sir. Now, it's my pleasure to welcome Dr. S.H. Nele, Laboratory of Medicinal Plant Biotechnology, College of Pharmacy, Chinese Medical University, Xi'an, on stage to add glory in this sensation and to develop the skill of excellency. And now I would like to request Dr. A.P. Pathak ma'am to please introduce our dynamic speaker, Dr. S.H. Nele, sir. Sir, please.
Once again, a proud moment for School of Life Sciences. I'm really pleased and feel proud to introduce a global scientist, Dr. Shivraj Nile. He is member of Life Science family in different and diverse capacities. First as a master's student, then PhD, then faculty member. And I feel very fortunate to have a sincere, bright, intelligent, and very kind alumni, Dr. Shivraj Nile. So it's my proud privilege, Dr. Nile. I appeal all student participants, please listen and appreciate his achievements. Now this is the CV, and this CV is really very impressive. Dr. Shivraj Nile obtained Masters in Biotechnology and PhD in from School of Life Sciences, Swami Ramananda Tirth Marathwada University. He has been awarded with KU Brainpool Postdoctoral Fellowship, Konkuk University. He currently working as associate and received RGNF Research Fellowship from UGC India. He has been included in top 2% scientist list of Stanford University. Then he has been awarded with 5 Young Scientist Awards from Konkuk University, Research Pioneer Award for his citations. His current citation is near about, it is equivalent to 4,500 with H index 31. His 7 papers have been cited for more than 100 times. Very impressive achievement. He had 10 years research experience in biotechnology, food science, phytochemistry, pharmacology, and nanotechnology. Dr. Nile had more than 100 research publications, out of which 8 papers with more than 10 impact factor. This is again very impressive achievement. 40 papers having impact factor more than 5 that include trends in biotechnology, critical reviews in food science, nutrition, then trends in food science technology, nano, many. I think this list is big. No, I have to read this to encourage students of life sciences, because these are the achievements of our alumni. So it is our right, and it is proud moment for School of Life Sciences. So I'll read all these: frontiers in pharmacology and food reviews. Then he he had been awarded with 4 patents and 6 research projects are in his credit. Then 15 national and international scientific research awards. He is working as associate editor for IJMS, South African Journal of Botany, Frontiers in Pharmacology, then food journal, some e-food journals, Journal of Recent Patents on Food Nutrition and Agriculture. One more achievement, which I have to mention, which is not given here actually. So one more very great achievement is his devotion plus his better half. She is also working as a scientist, and she has very impressive, I think more impressive work than Nile in cancer biology. So with this, I have to stop. And now I request Dr. Shivraj Nile to start his presentation.
Good evening to all of you. It's a great pleasure and very proud moment that today I'm presenting my research in front of my students, of my teachers and my friends. I've been here for at least 5 to 6 years as a master student, PhD student. Then I joined as assistant professor in Life Science, School of Life Science, and I got opportunity to work in South Korea as a postdoc fellow, KU Brainpool Fellowship. Basically, I'm working on herbal plants, food products, and their biological activities. Basically, cancer, gout, diabetes. Along with my, she's also working on cell biology, cancer biology, both working on antibody production against various diseases. So briefly, I will explain our research, not my research, our research, because we both working on different area, but like it's having same path. Next slide. So this is overall synopsis or abstract. Basically, for any natural drug, first we have to qualitative and quantitative analysis, and qualitative evaluation of herbal drugs is very important. For example, botanical characterization, physical characterization, chemical characterization, physical. So it depends on what parameters we want for the study. And I'm working on chemical and biological characterization. Then metabolic analysis, we are working on natural products, isolation, characterization by using different chromatographic and spectroscopic methods. So you people are familiar with HPLC, LCMS, and like NMR, all. Then we have to target like untargeted compound, targeted compound. Just now, Irshad and the sir, they are discussing about the compound, how they like screened. So we have to select targeted compounds like different flavonoids, phenolics. Next, after isolation and characterization, we are going for different biological activities. And for that, we are selecting 3 types of assays: in silico, in vitro, and in vivo. So the major difference is, first, in silico, by using computational methods, we have to check whether like particular drug is important for like particular disease, to control the disease. Drug score, drug likeness, the molecular modeling. Then in vitro by using different assay, colored assay by using different reagents, and in vivo means by using animal models. Next slide. This is like herbal characterization for development of food and drug. Basically, in our lab, we are working on functional food and drug. Now, in current scenario, that's very important. Food as a drug. So instead of going for like drugs which are having various side effects, so basically we are targeting food as a drug, and we are characterizing by means of chemical and biological analysis. Already I described in previous slide. Next. But we are not targeting only the plants. So simply cutting plants and making extraction and screening, that is not important. So we have to minimize the waste generated by food, like apple, banana, citrus. So we want to target, we want to isolate the compounds which are available in different waste. So we selected apple, citrus, banana peel, so different kind of fruit and vegetable waste. You can see how the waste can be used as a good source for humankind. Instead of throwing the food waste, we can utilize the food or fruit waste as a 3R: reuse, recycle, and reuse. So here we selected this fruit waste for phytochemical analysis and biological analysis. In different labs, they are working on even bioenergy production, also recycling and reuse for extract nutraceuticals and also agriculture products. So this is recycle and reutilization of bioactive compounds from fruit waste. Next. So we are targeting different cancer cell line in our laboratory. We are first selecting different cancer cell line, at least 5 to 6 cancer cell line, because it's very difficult to screen single cell line, because sometimes few compounds are very effective against colon cancer or some skin cancer. So we are selecting different cell lines for primary screening. Then after dose independent, then we are selecting that skin cancer cell line for further study. So we are basically, I'm working in China. China means people are afraid about COVID because COVID originated from China. But if you want to go for research in China, Korea, or Japan, so basically they have a lot of funding, and also they want people from India for as a researcher, as a student. So it's my duty to invite all students. Those who want to join China, Korea, I can help, because after this COVID pandemic, there are a lot of opportunities. Even in my lab, we have 9 positions, 3 postdoc, 2 PhD, and other masters. And for all these categories, you are getting very good fellowship from China, Japan, and Korea. So basically, for cancer, we have to target different pathway and different proteins, even cell death, delay or overcoming of drug resistance, regulate immune system. Because in during cancer progression, the immune system is very important. To enhance the immunity, we have to check whether our compounds having some immunosuppressor or immunoenhancer activity. Then we are targeting different proteins which are involved in cancer progression and development. So these are very common and famous compounds isolated from natural product, and the derivatives of these compounds were currently synthesized by chemical pharmaceutical industries, and they're utilized as a cancer drug. Next. So this is overall protocol, experimental hypothesis, extraction and analysis, and then activity. Here we also targeting different enzymes, the enzymes which involved in metabolic disorders. For example, xanthine oxidase, which involved in gout, the progression of, because the xanthine oxidase produce uric acid, and that uric acid get deposited in joints and it causes gout. So we have which are involved in diabetes. So even in cancer, there is proteins which can be down regulated and up regulated depending upon the protein function. So we are also inhibiting the enzymes from different sources by using these bioactive compounds. Next. So basically, cancer studies, nothing but we have to target different pathways, even cell cycle, then cell cycle arrest, then different protein regulation like BAX1, BCL2. For example, BAX and BCL2. One protein is anti-apoptotic and one is pro-apoptotic. So apoptosis is very important phenomena in cancer. So some proteins they are pro-apoptotic and some proteins are anti-apoptotic. So we have to inhibit that anti-apoptotic proteins, and we have to promote the pro-apoptotic proteins to initiate the apoptosis. Also, ours for cancer cell to enhance free radicals or reactive oxygen species are cancer cell development. Also, we are targeting apoptosis and cascade. These are the like different enzymes we have to target. Then membrane, mitochondrial membrane potential. That is also important to target particular site. Next. So this is one example. Now I'm like presenting one paper. One by one, I will explain. We extracted bioactive compounds from saffron waste, because China, even India, the like various Gulf countries, they are like huge production of saffron, and saffron is very expensive product in worldwide. But there is a huge production of waste, like different petals, sepals. Only we are using anther for saffron production all over the world. But what about the waste? So we collected the waste and we extracted bioactive compounds, quercetin derivatives, and also we are working on some sugars, because this saffron sugar, like petals and sepals, they have like sugar quantity. So even some polysaccharides are very like excellent source of anti-cancer agent. For any like development of disease or disorder, the main factor is sugar. Our body has sugar level, different sugar level. So that's very important. And some polysaccharides can be utilized as anti-cancer drug or also like antioxidant. So here we targeted different cell line. First, like we screened different cell line, primary cell line, and we selected MCF7 for previous slide. And along with this, we have to check whether our compound has cytotoxicity or genotoxicity towards normal cell. Here we are killing the cancerous cell, not the normal cell. For that, we have to check whether the compound like suppressing the cancer cell and enhancing the immunity, and also the genotoxicity is very important. So we selected LMCPA. It's a very simple experiment, genotoxicity, and we found that the extracted compound also, by means of flow cytometry, we checked the apoptosis, late apoptosis, early apoptosis, necrosis. So these are different stages of apoptosis. Necrosis means like there is already the cell death. So we have to check whether our compound is like targeting at which phase, late or pro. So that's very important, early or late. Necrosis means the cell already died. So then in genotoxicity, we have to check the alteration in chromosomal abbreviations like mitosis. You can see the metaphase, anaphase, and then we have to calculate the mitotic index. And depending upon that, we have to like prove whether our compound is having cytotoxicity or genotoxicity. And according to, we are developing the drug. Next. This is also like onion, and we found very excellent compound from red onion. And this compound now we are patenting in China. So now there is a big issue, because like I'm an Indian working in China, and my boss, he's from China. So he want that patent from China. I want that patent from India. So that is big issue if you work other country. So I'm trying to give my like potential to Indian institutes. So let's see. We extracted this like spiroside, and this is water soluble compound. For like drug development, water soluble compounds are very important compared to the solvent soluble, because these can be like in previous lecture, many scientists like discuss about bioavailability or targeted drug delivery. So we decided to utilize this extracted compound, and we synthesize nanoparticles, and also we are doing different like animal studies. Here we've found that spiroside is very excellent at very low concentration. For like good drug, the concentration of drug is very important for targeted drug delivery towards the particular cell line, like targeted cell line. So this spiroside we extracted, and you can see the protein expression, like beta-actin as a model protein, then mudeng, like it's a new discovery in Konkuk University. My wife discovered one protein which is anti-apoptotic, and that is first protein discovered by that lab, and they are now publishing maybe soon as a patent. And they got a lot of funding from Korean government for this mudeng. Mudeng is a protein which involved and which cause like anti-apoptotic property in various cancer cell line. And our lab developing one kit to detect cancer at early stage. So that is also, And for that, the Korean government, they gave near about $80,000 US funding to that project. You can see what is meant by protein upregulation and downregulation. म्हणजे जे महत्त्वाचे प्रोटीन आहेत त्याला अपरेग्युलेट करणे. Just we have to enhance protein expression, and downregulation suppress the protein, inhibit the protein. As I told, BCL2 we have to suppress, and you can see the BCL2 expression decreasing when we treated with the saffron at different level. First control 12 hour, 24 hour. So this is time dependent assay, and we found that spiroside is very effective against different anti-apoptotic proteins, and also regulating the pro-apoptotic proteins. Then we studied different extract along with, because sometimes the crude extract has maximum activity compared to the pure drug. So we have to compare pure drug and the crude extract. So we selected different extracts and along with the extracted compound, pure form compound. Next. So this is also similar study from apple, but I want to show one, like two compounds we extracted from apple pomaceae, and that is terpenic acids. Terpenic acids are very effective as a cosmetic agent, and we are targeting that compounds as a novel anti-cancer drugs. Next. Can you skip these 4 slides? These are like different carotenoids extracted from citrus, and we studied the same study anti-cancer activity by using HeLa cell line. So basically, I want to show the apple compounds which are very promising anti-cancer drugs, and we are getting a lot of funding for that, and we are collaborating with different companies. Next. So this work is very excellent because people just peeling the apple and they're throwing the cover pomaceae. But eating whole apple is very important for our body. Just we are eating the sugary part, not eating the peel. So we found that the peel contains very promising compounds, very interesting compounds, that is terpenic acids. And we extracted 6 terpenic acid. Here we selected 2 terpenic acids, UAL and erythritol. Olenic acid, uracilinic acid, these are the very important drugs already discovered by cosmetic industry, and they can be used as emulsifier fiber, like in your creams. So next, and we selected 3rd one is quercetin glucoside. The glucoside moiety is very important for attachment on the cell surface. Like quercetin cannot be easily penetrate into the cell surface, but quercetin glucosides are very attachable to the cell surface. Next. So these are 2 important compounds we extracted from apple pomaceae, and we studied cytotoxicity by means of time dependent and dose dependent. Time dependent means treatment with different time interval, like 12 hour, 24, 48, and we have to check the IC50, CC50. IC50 value in case of cytotoxicity, CC50 value for normal cell line. And we found that these compounds are very excellent compared to the available commercial drugs. So this is cell cytotoxicity assay. Next. And this is flow cytometric analysis, cell cycle analysis. And here we want to target G2 M phase or S phase, like synthetic phase, where the molecular compounds were synthesized. So we have to target cell cycle at particular phases, G2M or either S phase, synthetic phase. So these 2 compounds are very excellent to target the cell cycle, like mitosis in cell. So you can find in these 2 compounds targeted G2 and M phase at very good concentration. Next. Then this is time dependent assay. It's similar like dose dependent. Here we're considering time, and in dose dependent we're considering the concentration. Next. This factor is very important, mitochondrial membrane potential, because in cell the mitochondria is a powerhouse, and we have to break the membrane of mitochondria and to release the different cytochromes, and which are involved in progression of cancer cell line. So We studied the effect of these drugs on mitochondrial memory potential at different time-dependent assay. And we found that these drugs are very excellent to break the mitochondrial membrane and to release reactive oxygen species, because reactive oxygen species are very important to to control the proliferation of cancerous cell line. Next. So you can see that chromosomal DNA degradation and inhibition of colony formation. So this is like simple assay colony formation by using two compound at different time interval. And you can see at 48 hour, there is, you can see the number of cell or cell viability is very less. So formation of colony is very less. So it indicates that our two compounds are very excellent to inhibit the colony formation of cancerous cell line. And finally, we did the DNA, chromosomal DNA degradation assay, and we found our compound like very helpful to degrade the DNA which in present in cancerous cell. Next. Then autophagy. These are different parameters in cancer cell progression. So autophagy. So we also studied these compounds are very important. That's in autophagy. These drugs like inducing the autophagy cell to go the under lysis. Next. So this is like signaling pathways, because in cancer cell or in cancer development there are different signals. Protein provides signal to other protein for progression of cancer. So this is MAPK activation triggered by erythroid UOL, and also ROS production, autophagy and apoptosis induced by UOL and erythroid. And we selected different proteins which are important for like cell cycle, this signaling pathways. And you can found like P53.8, these are different proteins involved in, like these are the cascade of proteins which involved in cancer cell development. So we targeted these proteins by means of our compound, and we upregulated the important proteins and downregulated the like anti-apoptotic proteins. Next. Then molecular crosstalk between so these compounds have some like talk between different proteins, and they are targeting we are targeting different. So this is also same different protein, and we are targeting these proteins by means of our compounds. Next. So this is all about like pure compound. So what about the like targeted drug delivery? So we selected one nanomaterial synthesis phenomena, like utilization of nanotechnology. Here combinational therapy, like for example आपल्या क्लासमध्ये असताना एक हुशार मुलगा असेल तर एखाद्या बॅक साईडच्या मुलाला म्हणतात की हुशार मुलासोबत बस. सेम वी आर डुईंग हिअर. For mycophenolic acid is very strong anti-cancer agent, but it has very like high toxicity, and quercetin has less cytotoxicity. So we selected these two compounds and combined by using liposomes, and we targeted on different cancer cell. You can see the in case of pure drug, single drug, the mycophenolic acid, the metabolism is high, then viability is less, pharmacodynamic action is less. But if we combine quercetin and mycophenolic acid, the metabolism reduced, because quercetin block cytochrome enzyme, cytochrome 450 enzyme, which involved in metabolism of mycophenolic acid. So metabolism decreased, the viability increased. So you can see the tumor size just beside that one, tumor size. The combination of two drugs along with liposome, we combine two drugs by using liposome nanomaterial, and you can see the size of tumor compared to the control is very less. So it means that the combinational therapy by using nanotechnology is very impressive compared to the like single isolated drug. Then we also checked the size, as Kharasar told, size, nanomaterial size is very important. 10 to 100, even like 10 to 300 also like permissible in research. So our compounds are like 200 nanometers. By using zeta potential, we calculated size and all things. Then drug effect and cytotoxicity we selected. Just we can say that combination therapy of these two drugs by using liposomes remove the hurdle of individual drugs and improve the therapeutic efficacy. Even it gives higher cancer cell cytotoxicity completely. Also enhance the anti-tumor efficacy by like reducing the tumor size. So this is all about nanotechnology. We are also working on COVID-19. We like screening drugs by using in silico screening, and we got a lot of publication on COVID also. And also we are working on influenza virus drug development. In like China, Korea, they want product or process. So just sir discuss about process patent and product patent. So in China, if you want to survive in other country, you have to give your targeted work. We have to finish within time, at least one patent or 3 or 4 papers in a year. So these are all work related to our research group. Next. Next. So we have like extracted compound in very small quantity. But what about like if you want to go bulk, then we developed hairy root culture in our laboratory. And for this paper, like this paper published in Trends in Biotechnology, like impact is 19. So hairy root culture is very important to enhance production of metabolites. Now we have metabolites, but the concentration of after extraction, we are getting very less concentration. So we want to enhance the production of that metabolites. That's why we developed hairy root culture in our lab. In our lab, we have 40 students and we have 3 groups: pharmacology, nanotechnology, and metabolic engineering. So 3 different groups, they are working on different aspects, and we are doing like collaborative work in our lab. So hairy root culture, we utilizing, and we are going like producing chemtothecin in China, because Chinese government, like Artemisinin, one lady from China, she got Nobel Prize on anti-malarial drug. So China want such kind of work from Chinese people, and they are giving a lot of funding. So chemtothecin is a very good drug, like anti-cancer drug. And our lab is first lab. We are like doing hairy root culture for mass production, and we develop different technology. Even we are comparing different bioreactors to enhance production of these bacterial compounds. And we after that we are going for like nanomaterials by using liposomes or encapsulation. So these are very important. Nowadays, nanotechnology is very important for targeted drug delivery. So this is all new, even biomaterials, by using gene protein, like maize protein, which can easily bind to the cell surface. So this is all about new research, and we are like for this I got funding from university, even National Science Foundation in China. Next. So this is my lab. You can see different postdocs and professors, and only 2 Indian guys like Gopal Patel. I invited Gopal Patel last year, and this is our university. It's in like very remote area. So you can see my professor, our lab boss, Professor Kai, and all other like Chinese people. Just I want like one of from your like from your like our department should be there like in photo in future. Thank you very much.
Definitely, sure, sir. One of us, we will be there. So thank you for your informative talk. And the basic is how waste can be converted into wealth. So waste to wealth is the tagline of your work, sir. Thank you very much. It's a multi-dimensional study of perfect example. And my words are very less. So thank you very much, sir. Now I would like to request Professor T.A. Kadam, sir, to please felicitate Dr. Shivraj Nile, sir. Thank you very much, sir. Thank you. Now, the next talk is Now, the next session is Dr. K.D. Sonone, sir, who is joining with us from Kolhapur in Structural Bioinformatics Unit, Department of Biochemistry, Shivaji University, Kolhapur. I would like to request Professor T.A. Kadam, sir, to please introduce our next speaker, Dr. K.D. Sonone, sir. Sir, please.
Welcome, sir.
Thank you, sir.
Since from 2:30, sir is waiting for presentations, and actually we are running late, so it was difficult to give presentation to the sir. So I briefly introduced Professor Kailas Sonone, sir. Actually, I received 22 pages CV. So now it is very difficult to introduce Kailas Sonone, sir. But briefly, within 1 minute or less than 1 minute, I will introduce Professor Kailas Sonone, sir. Is presently head department of microbiology and coordinator PG diploma in bioinformatics, Shivaji University, Kolhapur. He did PhD from NCL, that is Pune University, and MSc from Shivaji University, Kolhapur. Research specializations, bioinformatics, computational biology, antimicrobial peptides, and antibiotic resistance. He got fellowships from different institutes, reputed institutes. Example, National Institute of Health, USA. He has more than 24 years experience. 11 students have completed PhD under his able guidance. And presently 7 students are working under his guidance. He has experience of editor board members of many journals, notably Journal of Bioinformatics, Computational and System Biology, Precision Medicines, Journal of Enzymology and Metabolism, the Journal of Microbial World, member of professional societies like American Chemical Society, Royal Society of Chemistry, Biophysical Society of India, Association of Microbiology of India, Microbiology Society of India, 2 books on his credit, and 4 biological database he has developed, more than 75 research publications at international levels. Actually, more than 20 pages CV is there. So I will not take more time. I request, sir, please start your presentation, sir. Thank you, sir.
Hello, am I audible? Hello.
Yes, sir. Yes, sir.
All right. Okay. Hello. Is my screen is visible? All right. Okay. All right, then actually, first of all, thank you very much for all the organizing committee members for giving me this opportunity to talk in this particular conference. So at last, I got time slot to talk in this particular conference. So thank you very much, all the organizing committee members. So my talk would be on proteomics approaches to understand multi-drug resistance microorganisms. Find out. Hello.
Yes, sir, you are audible.
I am audible. Okay, because sir, इथे पाऊस पडतोय आणि that's why maybe disturbance is there.
Okay, okay, sir.
All right, okay. All right. So not only that, molecular docking to understand protein ligand interactions as well as drug receptor interaction can also be useful in this particular scenarios. So the data obtained from the functional genomics and proteomics could be useful in drug designing and discovery process. And the next approach that we can call as a proteomic approach that we use in order to understand this antibiotic resistance issue. So these slides are just for student purpose, as I said. For example, how the primary structure or linear sequence of any protein or any enzyme which is responsible to cause the resistance to the particular antibiotic. That can be downloaded from the NCBI protein sequence database. So here one can see the greater than symbol, GA number, and primary sequence of the protein. And all the such databases can be obtained from the primary databases. And the program that which we use for the molecular docking, that is an automated docking software for the predicting optimal protein ligand interactions. So this is a very useful technique right now. Most of the pharma companies, as well as research institutes, universities, colleges, several colleges, and staff members are being used widely to understand how one particular drug molecule interacts with the receptor molecules. And with respect to that, there are two approaches. Like if we have a protein as a receptor as well as a drug molecule, then one can try to find out if the any ligand as a protein molecule, if it fits properly into the receptor binding domain, then that would be considered as a proper binding sites. But however, one has to perform further energy minimization, molecular docking studies can be performed. And then if we have a small drug molecule, and then this is the receptor kind of a thing, and this is a small drug molecule. And then also one can try to find out if we do not get proper interactions, then that molecule has to be optimized further. And then we can get minimized structure that we can call as optimized conformation of that particular drug molecule, which is interacting with the receptor molecule. And that's why this field called as a computer-aided drug designing is a very important area right now. And most of the pharma companies particularly, they are interested, they are working in this area in order to reduce the time period which is required if you go by traditional drug discovery process. And that's why if we could use these particular methods, one could discover any particular drug or get that drug into the market within maybe 4 to 5 years of time. And that cost of that drug would also go down, so that the additional burden that every like users like we all would also, that cost of that drug would also go down. That is the advantage of this particular methods. So the computer-aided drug designing is a specialized discipline that uses computational methods to simulate drug receptor interactions. And the CADD methods are heavily dependent on the bioinformatics tools, applications, and databases. As such, there is a considerable overlap in CADD research and bioinformatics activities. And this is the important technique that we can call as, I will be talking on this particular method, that is the virtual screening for a drug discovery method. So what we do here, I think again connection has lost. I hope I am audible to everyone. All right, so antibiotic resistance among various infectious agents towards different types of antimicrobial drugs has emerged as a global cause of public health threat. Antibiotic resistance is the ability of a microorganism to withstand the effects of antibiotics. So the diverse newly developed resistance pattern among microbial species and the decrease in the efficiency of treating common infectious diseases resulted into the development of high levels of multi-drug resistant strains. And that's why the resistance of this particular antibiotics with respect to aminoglycoside group of antibiotics, which we are working on, particularly streptomycin, gentamicin, kanamycin, penicillin, all these things, mainly occurs by the aminoglycoside kinases and thus targeting AKs.
Sir,
Yes, sir.
your slides are not visible.
Okay, okay. Because somehow I lost connection, and let me share again. Okay, are they visible now?
Yes.
All
Yes,
right.
sir.
Okay, sir. Okay. So let me repeat with the earlier slide. So the last para, that antibiotic resistance to aminoglycoside group of antibiotics mainly occurs by the aminoglycoside kinases. And basically these aminoglycoside kinases are also expressed in human cells. And some bacterial systems we can also express. So there should not be kind of a cross connection between these particular, and that's why the resistance occurs. So thus targeting AKs from different multi-drug resistant strains could result into the inhibition of AK, that is aminoglycoside kinases, and AK enzymes, and ultimately the resistance. All right, so there are several mechanisms of this antibiotic resistance. Let me share some light on this particular thing. If you see this particular A, that is alteration of the target site by the antibiotics. So bacterial system can target the alteration of this particular receptor so that that antibiotics cannot be effective. If we come to the B, so that there are certain enzymes that can be expressed by the bacterial system as soon as they come in contact with any specific antibiotic. So those enzymes can deactivate the particular enzyme, particular antibiotic, maybe by addition of any additional groups, particularly maybe phosphate groups, which we are going to talk in this particular talk. All right. So this enzyme inactivation can be done by enzymes secreted by the bacterial cell, and then active transport of the antibiotic out of the bacterial cell. So some antibiotics, they just go, throughout this particular membrane. Okay, again, I think there is connection is coming up and down again and again. So let us begin with this one. So, okay, so this is the earlier part that which we had published in Journal of Cellular Biochemistry, considering the molecular docking approaches and molecular modeling approaches and virtual screen inhibitor towards the different. Yes, sir. Yes, madam.
जर पुन्हा डिस्कनेक्ट झालं ना, जर पूर्ण झालं तर ठीक आहे. कनेक्ट झालं तर परत लॉग इन करण्यापेक्षा उद्या दुपारी आपण घेऊया, चालेल?
मॅडम, उद्या दुपारी होल डे आय विल बी बिझी ऍक्च्युअली. सो मे बी लेट अस से जोपर्यंत होतो तोपर्यंत कंटिन्यू करूयात, कारण इकडे वातावरण थोडंसं आहे खराब झालं.
हा, म्हणूनच उद्या म्हटलं होतं.
नाही, उद्या होल डे आय विल बी बिझी इन सम अदर ऍक्टिव्हिटीज ऍक्च्युअली.
फाईन, जेवढं होईल
सो लेट
तेवढं
अस
करा.
सी कुठपर्यंत जाते बघूयात आपण ते.
ओके.
All right. Okay. All right. So in this particular work which we have done, that is particularly identified one drug molecule that is called as with this particular number that is called as the zinc ID from the zinc database. All right. So initially we had performed sequence alignment that we can call as a multiple sequence alignment with the AKs or amino APH enzymes from different organisms, particularly to this Mycobacterium tuberculosis.
So good evening, everybody. I'm here to summarize this session. Actually, I'm not indulging in much time. We had a very busy session actually, but it is very actually fruitful session. Who witnessed it? At the onset of this session, Dr. Pradeep Paik sir, he discussed about the polymer-based national, what you call the nanotechnology and biomedical biomedicines. Then Professor Venkateshwar, he discussed about the mitochondria as the new target as he identified for this what you call the dengue virus. Then Professor Radhesham Maurya sir had a very excellent talk, had a very heavy discussion about his topic in this regard with the special reference to Withania somnifera and its use as a drug. And he explained all the biotechnological approaches as well as how it can be eligible for the international patent. Then we had a talk of Dr. Shivraj Nile at the last in this session, who remained the alumni of this university as well as my best student during his BSc. He discussed about use of the waste, fruit wastes, to have the bioactive compounds which can be utilized as the better drugs against the cancers. He and his better half, Arti Mahajan, they were working on it. And lastly, we were trying hardly to listen Dr. K.D. Sonone sir, Kailash Sonone sir from Kolhapur, but unfortunately due to the heavy raining and thunders in the Kolhapur, the continuity of what you call the internet connection is not there. He will discuss his topic in the morning in 9:30 as per the schedules tomorrow. Kailash सोनोने सरांचं उद्या सकाळी 9:30 ला कदाचित होईल इथल्या मॅनेजमेंट प्रमाणे. So thanks for listening. Thanks to all. Thank you.